आप सभी को मेरा नमस्कार। ज्ञान गंगा कार्यक्रम के चतुर्थ दिन मैं डॉक्टर नितिका सिंह आप सभी का हार्दिक स्वागत करती हूं। आज के हमारे कार्यक्रम की थीम है एप्लीकेशंस एंड रिसर्च मेथोडोलॉजी इन एल्गी। तो ज़्यादा समय न लेते हुए हम आज का सेशन प्रारंभ करते हैं। नाउ आई वुड लाइक टू इंट्रोड्यूस आवर फर्स्ट एमिनेंट स्पीकर, Dr. V. Siva Subramanian. Sir serves as Director of FICO Spectrum Environmental Research Center, Chennai, India. He has spent more than 25 years in teaching and in acquiring research experience in the area of algal technology. Sir has received the PhD degree in algal physiology and biotechnology during 1983. Since 1996, Sir has been actively involved in developing algae-based technology to treat industrial effluents and wastewater and implemented large-scale treatment systems all over India. He is a pioneer in taking algal technology to handle effluents in large-scale industrial treatment plants. World's first FICO remediation plant was set up at SNAP Alginates Industry with the research support from his group. Sir has guided 20 PhD students and handled several industrial projects and published more than 100 research articles in national as well as international journals. He has served as the core committee of experts on second generation biofuels, Ministry of New and Renewable Energy, Government of India, and member of monitoring committee of CSIR and DBT for projects on biofuels from marine microalgae. He is the chief editor of Journal of Algal Biomass Utilization and a member of Review Committee of Bioresource Technology Journal, member of editorial board of Seaweed Research and Utilization Journal, assistant editor of FICOS of Phycological Society India. Sir has got many prestigious awards like Best Reviewer Award 2014 for Bioresource Technology Journal. Sir is in collaboration with Core Biotech to implement FICO remediation in Latin America. Iran's first algal cultivation facility at Chabahar has been established in May 2015 with technical support from Dr. V. Siva Subramanian. He has recently signed an agreement with Astral Industrial Corporation Limited to put up algae production facility at Sicily for biomass to biogas production. Sir is also involved in a project to restore a drain in Gulati, UP in National Mission for Clean Ganga. Today, Sir is going to deliver his talk on microalgal technology for effluent treatment and restoration of polluted water bodies. I welcome you, sir. Please start your presentation. Dr. V. Siva Subramanian.
Thank you. Okay, I'll start my presentation.
Sir, please share your screen.
Yes. Is it visible?
Yes, sir.
Okay. Then full screen?
Yes, sir.
Do you see the title?
No, sir. See the title.
No? Right. Now?
Yes, sir.
Okay. I was sharing something else. Sorry. Okay. Good morning, everyone. And it is really happy to be here talking to people from Rajasthan. In fact, I was traveling in Rajasthan 2 weeks back. Do you hear me? My sound is okay?
Yes, sir. Yes.
Okay. So last 2 weeks back, I was traveling in Rajasthan. So we have projects in Kota and also in Jaipur. In Jaipur, we visited the sewage treatment plant in Kota. I mean in Jaipur, where we have introduced our RL technology, and I will be talking about it later if there's a time. And we also have pilot plant, algae-based pilot water, DCM Shriram Limited in Kota. I think somebody's in the audience. I saw somebody from Kota. So I was there in Kota 2 weeks back, and I've been traveling Kota even before the COVID period. Because of the COVID, we couldn't come there. So the first visit was 2 weeks back. And my topic today, I think you can see it's about the microalgae technology for treating industrial wastewater and also restoration of polluted water bodies. So I'll be including a lot of things. So I'll be very brief to tell you what we have been doing for the last 20 years. So I may not give you the entire presentation, and because it is only 45 minutes. So I just started 10:38. So I'll be stopping at 11:05. So we can, I think, can have 10 minutes of questions. After that, 11:15, I think I'm supposed to stop. So I'll be starting with this first slide. So you can see here our collaboration with Colombia. So we have a big collaboration with Colombia and the company called PICOR. The PHY refers to Picospectrum, our center, and core refers to the Colombian company. And PICOR is a joint venture company. So we are going to establish a big plant, a power plant that will be treating algae, and algae will be grown in sewage and wastewater, and algae will be used to produce biogas and power. So this is, I think I'll be explaining it later if there is a time. And this is our team. I was talking about our first project in Colombia a few years back, where we have been asked to put up an algae-based plant to treat the wastewater from petroleum industry, the petroleum industry in Colombia, the second biggest. And this is our team, and this is me. And we used to go there, and I used to stay for a month. And very recently, we just we started a company called PICOR and established it with my partner, who is a doctor, a medical doctor, and he is owner of the Core Biotech, and we jointly call our company as PICOR. And this man is now doing his PhD in algae in Bogota University in Colombia, um, and he'll be submitting his thesis in algae in a couple of months. So this is another project that we just started in Colombia. I think this map. You can see a small city here. It's called Puerto Colombia, and this is the Caribbean Sea, and this goes to Barranquilla, and this goes to Cartagena, and this has a sewage treatment plant here. It is enlarged here. Just to show you the details, and we have actually isolated microalgae from all these three ponds. With the sewage treatment plant, all the three ponds, they have different composition of algae. I think we are studying on it, and we'll be entering into agreement with this city to treat this. And we have done another project in Colombia, a Camagüey slaughterhouse in Barranquilla. So where we have introduced one of the reactors called BF plus. I'll be explaining this later. What is BF plus? So this was done very successfully, and we have treated the effluent, and the biomass is now used for, and we have this bigger version of it coming up in Colombia. bigger version, and we are actually treating the effluent and then making biomass. I think you see the biomass. The normal brown coal has 3.5 calorific value, calories per kg, and industrial effluent biomass has got about 3.4, 3.8. Domestic effluent is about 3.8. So we are able to get the biomass with a higher calorific value in Colombia. So this is the very latest picture that you are going to see. We see the video running. You can see the map, Google map, which has got a video. Do you see it is loading? Okay. So now you can see last March I was there just before we shut down for COVID. I was there in Colombia. So this is the canal here, and that goes to a big swamp here. I can see a big swamp, and this big swamp. I'll show you the next picture. Yes, this is where we started, and this is our team there, and we have a very big team in Colombia, and we have also we are also involving the school students there, and because this is a small village nearby and there's a very small school, and we have included senior students from various classes to form a group, and these girls who are having this green t-shirt, they are the leaders of groups. So that we have found about 4 or 5 groups, and these groups are named after one each alga. So I was there in large March uh, to train them, and this is the entire team of school students. They will be making these charts, and this is school, this teacher of these students, and we are actually encouraging the students to study. And they have included phycoremediation, algae-based technology for effluent treatment, their syllabus, and they are teaching students the basics, and they'll be making charts and the other materials that will be distributed, and they'll be explaining them to the local people there. So we actually met these people, local people, to tell them what we are doing. Because in Colombia, first you have to convince the local people what you are doing. Then only we can proceed with whatever we want to do in large scale in the water body. So this is actually the facility that we have in Colombia, and this is a little far away from the civilization. You can see it is nobody is there, and we have a facility where we are actually growing. This is supposed to be the world first large-scale phycoremediation production site, where we will be uh, growing algae collected from various sites, and then produce the biomass, and take the biomass in yes, you can see the facility here. And we take this algae in this, I think you can see here. This is in Spanish, don't worry. So we are using this tanker. It's an aerial view. So we have a huge facility in Colombia where we will be producing the algal biomass, and then take it in big tankers and then put them back in the wastewater. The idea is, there are two types of algae, the beneficial algae and then harmful algae. So we try to minimize the harmful algae by increasing the number of beneficial algae by collecting them and putting them in a culture facility, a growth facility where we dose a lot of materials, nutrients, and then grow and then take this concentrated biomass, put them back in the wastewater, where they will be fighting with the, the the harmful algae, and also taking up the nutrients very effectively. So we also had on March 4th in 2020, that's before we shut down for COVID, we had a press meet, the TV people, the newspaper people, all of them. I mean, whenever I go to Colombia, this happens. So we have a laboratory there in Barranquilla, in Colombia, and we are growing this algae collected from the various sites. And this is our collaborators. And we also received, as you can see here, Latin American Green Award 2020 Sucre in August 22nd, 2020. When we were sitting inside our homes for COVID, we have been given by the Latin American group because there are about 24 countries in Latin America, and about 4,000 the projects have been submitted, and we have been given the award for Green Award for 2020 for a project we have done in Sucre. Pico means Pico, and we are the winners. And after we became the winners, we have become popular, and a lot of people started visiting our facility, and the mayors and the local people. They, so this is a TV commercial. So this is when the mayor, is the mayor, governor of Sucre, when he visited our facility, this came in the news. So this is a news item which came in the TV. So governor himself is explaining our project there. And another project we just started in Colombia is swamp restoration. So this is a big swamp you can see here. This is the Barranquilla city. It's a big swamp, Malacan swamp, and this is the Caribbean Sea, and this is a river, the big river, Magdalena River. So this is situated in between the Magdalena River and the Caribbean Sea. It's called as the Malacan swamp. So this swamp is now being cleaned up, and we started our project already. And this project has been very successful, and we also started cleaning up the small waterways that goes and dump their waste using our technology. And this came in the newspaper also. And we have been given a big project now to maintain this swamp. And this swamp is getting, promoted into a travel or a place for spending time and all that. So this is the news item that came in the local newspaper. And this another project we just started in one of the industries in Barranquilla, called as Canon textile industry. And I visited on 4th March. And this is our reactor. The importance of the reactor is this is a continuous process, first time in the world, and very good addition, BOD, COD, color, and toxicity. You can see the various sample. This is a raw sample, and when it goes through the system, at the end you see it is almost transparent, and all this BOD, COD, color, toxicity have been reduced to very low level. So this for the first time, I think we have made a continuous process. Otherwise, you will be doing only semi-continuous process. I'll be explaining this later. There is a time. And this is another project we just started in Iran to produce tunalial, and we are also involving local industries to supply their flue gas so that we can grow this biomass very effectively. And another project we just started in Welsh, in UK, where we have this partner. This is Dr. Tajuddin from India, and this is me, and this Carol and Ella from Swansea University, and they are with us, and we'll be taking up this project for wastewater. Wastewater treatment using microalgae, and this is another visit we made in Port Talbot Steelworks, Tata Steel they have, and after coming back from to India, I was invited by the Tata Steel company to look into their problem in Jamshedpur. They have a big problem of color, cyanide, and ammonia removal. And this removal, when the sample was sent to us to a laboratory in Chennai, we were able to remove the color and bring down cyanide and ammonia within 4 hours. So this is after treatment of 4 hours. And this is the pilot plant that we have just started there, and it is almost under completion. And this is where we are going to scale up. And this is the aerial view of the sewage plant. It's a treatment plant, wastewater treatment plant. These are all railway tracks there inside. And this is where we are going to intervene. And this is a big system, about 8,400 meter cube, huge system. And we made a smaller version of this big system with all the uh, pumps and then all the inlet and outlets. And we have also fixed lights, artificial lights, and this is the 8,000 liter pilot we have completed, and it was really successful. In fact, we did another project in Tamil Nadu, in Titan, where we were able to treat the airplane that has got about 58,000 TDS, and the TDS and ETP goes up to 65,000. And it is an operation process where we are able to remove the TDS, and this is the scaled up plant we have put up in Titan, the jewelry division, and that part of Tata company. And these latest pictures that you are seeing here. And this is another uh, trial we just started in 2019, I think December, in Tata Steel Jam in West Bengal, not in Jamshedpur, West Bengal. This is a 40,000 liter pilot plant we have started. This is an evaporation surface, surface 1 and surface 2. And this is evaporating about, we are actually adding 5,000 to 10,000 liter to the 40,000 liter tank here. And the process I will explain a little later. And we are also working on the other projects like lake, pond, and also river and stream and other evaporation or treatment in India, also in other countries. So this is basically the algae power. The diagram explains it oxygenates water, eliminates odor, and degrades organic inorganic pollutants, and eliminates all harmful bacteria, and promotes biotic life in the natural way. So this is what we are actually putting in natural water bodies, because when you compare natural water body and the industrial effluent, the treatment is completely different. For industrial effluent, we give 99% artificial and 1% nature. Whereas in a natural water body, river, pond, or lake, or swamp, we give 1% artificial and 99% nature. So this is the basically our technology. We have developed a product called as Phycoplus. It's a mixture of organically derived nanotech paste, nutrients, trace elements, growth factors, and enzymes that actually, when it is applied, it enhances the production of algae and also bacteria. The bacteria are needed to degrade the organic load, and they are mineralized, and the minerals are taken up by algae. And algae, by using light and the taking up the nutrients and fixing carbon dioxide, it's evolving oxygen. The oxygen is taken up by bacteria. So this is a cycle which you are talking about in the technology that we normally use. In lakes, normally you will be seeing bacteria growing large number, completely covering the surface and cutting the light. I think, and they also produce siderophores that will scavenge iron, and iron scavenging actually makes cyanobacteria dominant and makes the prevents other algae to take up nitrogen. So nitrogen is completely cut off because of this scavenging, iron scavenging by cyanobacteria. And we actually, this is how it looks, right? The polluted lake, you'll see cyanobacterial and duckweeds, and they cover the entire surface, and light is not penetrating. That means low light, water appears black, and high BOD and COD, and high coliform bacteria. Water becomes black, and nothing grows except the anaerobic bacteria. So we actually remove this cyanobacterial and duckweed mat by our technology and allow the light to penetrate. Once the light penetrates, water becomes transparent, and that becomes low in BOD and COD. And they optimized its all oxygen, and no coliform bacteria and other harmful bacteria will disappear. And normal food chain is actually restored. So this is what we do. This is what we started in North India, near Aligarh. We have been given a project to restore a lake. It's a lake, you can see the screen, and when we start adding our product along with microalgae that we are growing there, and with our Phycoplus, I think you can see this before and after. And before, it's completely covered, the water is black and whole smelling. And after a week or so, you can see the water becoming transparent. You can see the bottom of the lake. The cyanobacterial mat is completely disappeared. And if we see the parameters, you see the POD, COD, and especially E. coli and total coliform bacteria are completely gone, and water becomes transparent and restored. So this is what we have actually demonstrated in Aligarh, near Aligarh, and this is for taking up a project under, Ganga mission. But what the Ganga mission people said was, okay, you have done something with the pond, but then Ganges, we're talking about running water. So we did other projects in temple tanks, and also we got the project from National Mission for Clean Ganga for treatment of Kulauti drain in Kulauti. And this, for the first time, the technology, PICOPS technology has been approved by the Clean Ganga Committee. And we started this in this village. And these are all other projects that we started in uh, other places. You can see in Bihar, we have restored a polluted lake, and the fish are dying due to disease and all that. We about 15th day, you can see the water becoming transparent, less greenish, and the fish started in, moving very well, and the disease disappeared. This is another project we did in Bihar, in Sitamarhi, Jangi Pond. And this is another project. You can see within 15 to 20 days, we are able to restore these ponds with the treatment we have for what we call as backup treatment. And this is another project we just started just before the COVID. This is a project in Meghalaya. You can see the Meghalaya, the rivers, most of the rivers are polluted by the mining activity that is going on. You can see a lot of mining activity and the coal mine and the sulfuric acid that is produced in the mining activity that enters into the river. And the river, you can see the sulfur coating on the rocks, and the water becomes transparent because nothing grows there, because the pH is around 2.5 to 3.5. No, nothing grows. So we have been given one of the rivers to restore. We just started a facility there. You can see with at night these recipe pond facilities, with artificial lights, and it is daytime. And we started this project just before the COVID, and it is going on. And we started dosing on the 10th November. And this is what the check dam we have created 3 or 4 places to grow algae, and we introduce algae into this. And the check dams created at 4 locations up to 1.2 kilometer and closing about 7 lakh liters where we'll be developing microalgae. And this is a little older, this thing. And we also encourage, because the pH has been corrected to more than 7, and we encourage the local people to come and wash their clothes. Otherwise, they will not come to this water out because it's very acidic, 2.5 to 3.5. Now, the water has become more than 7, and we encourage the local people to come and wash their clothes. We supply them with soap so that the rest of the water in the lower end also becomes utilized. So this is the latest picture that I can show. And we also restored, before we got this Ganges project, we also restored a river in Maligaon called the Mosam River. And we actually engaged with the UT Textiles on near the river, and actually they have been dumping their waste, textile waste, without the treatment. For 1 minute. Wait, பண்ணுங்க. Okay. Without any treatment. So we have engaged with them, and we have put up a large-scale system for treatment. And this treated water along with algae is let into the river. And you can see the table here. I think COD, this table is before and this is after treatment. You can see COD after 10 kilometers. So this is the 0 kilometer, and this is the middle, this is the 10th kilometer. Of the 10th kilometer, the river actually joins under big river. And COD at 10th kilometer is 188 before treatment. And after treatment, the COD at 10th kilometer becomes 21. So that's very good treatment. So the moving water actually carries the algae that we are adding. And then when the algae is carried, the treatment happens. The more distance it carries, I think it will be more effective. So we are able to follow up to 10 kilometers. At 10 kilometers, you can see the 188 COD becomes 21. That is excellent reduction. And we submitted this to the people, and we got project in the Kulavati. And this is another project. I think I told you about the Pacific Energy project that we did in Colombia, and this is the aerial view of the pictorial view of our plant, and this gives an idea about the continuous system of tanks that we are using. And this is where we did our pilot, and this is our group, and this is the effluent from the petrochemical waste. So we were able to reduce the parameters, especially the coliform bacteria and then the nutrients, and we started dumping it into the river. And we have another project that we just started. I'm not able to travel in Turkey. This is biomass from chicken waste generating around 200 TPD digestate. So digestate is basically used by algae to grow, and then we will be actually treating the waste and then producing algae, and algae goes back into that will go back into the digestion unit where they will producing power. And this is another project we just started in Indian Oil Corporation in North India, and refinery. I can see the raw effluent, the algae in the algae reactor, and the settled biomass of wheat. So this is another project I told you about my visit to Kota. In Kota, in Shri Ram Industries, we have started a pilot there. This is in night, this is during daytime. About 10 to 15 pilot. We have improved this to pilot about 15 KL, where we will be handling ammonia, COD, BOD, sludge, etc. So we have partner Trinity in New Delhi. So both of us joined together to do this project, and through all some of the pictures, I think it is 11:04. I can stop here and ask people to the shoot questions. We can answer. So in the meantime, I'll be just going through rest of my slides. So these are all some of the companies. Hello, you can ask questions. You have another 10 minutes to close.
एक मिनट.
Am I audible?
Yes sir, yes sir.
Okay. I think I completed very quick. I don't know. It is already 11:04.
I request to all the participants, please ask your question.
Because I have completed only a few slides. Actually,
Okay, sir. Sir, please unmute your mic. If any. Sir, please unmute your mic, sir. कैसे जा रहा है?
Oh, now you can hear my voice. Hello. Do you hear me?
Yes, sir, you are audible.
Okay.
Sir, you just please continue with your lecture, and after finishing that, we will take questions.
Don't worry, it is 11:05. I have completed how many slides? 100 slides. I have 240. Don't worry, I cannot complete all that. I will wait for questions, and I will just go through the slides. I just put the slides so that if anybody wants to ask questions from the slides, they can always ask. So these are all the benefits and advantages, and we correct pH, we correct color and odor removal, we reduce sludge, BOD, COD reduction, nutrient removal, We're doing ammonia, arrow reject management, and reduction of operation cost. In fact, on 90% reduction, because we are not using chemicals, and of course, because of flue gas and carbon mitigation, we are using oxygen walling of algae that will actually take care of the carbon. So this is our first project. We stayed some 15 years back in Tamil Nadu, where we started the work, and after that we entered into agreement with a lot of companies in India and also other countries, and the major countries are the Sweden, Turkey, and Italy, and major projects in Colombia. So we have a joint venture company in Colombia. You can ask your questions. I'll just go through the slides. If you want to pick up anything from there, and this is one of the first projects we did in India with algae. We actually make a lot of products out of algae, like the plankton, the biofertilizer, EMI, another fertilizer, and fish feed. And we actually, this is just to show you, I mean, how before adding our product and after adding our product, the polluted algae that disappears, and you can see the plankton actually helping improving the DO, zooplankton, phytoplankton, and we are tested all over India, and this is how it looks after the treatment is over, and we also remove carbon for the photosynthetic activity of algae that we are putting in the treatment plant. We are able to remove in the first project in SNAP in Tamil Nadu, we are able to 1,200 tons of carbon every year. So this latest picture that I took last month, that is 8th January, I was there. They have improved the whole facility. I will explain this if somebody asks a question. So we go on improving the facility that we have put up. So this facility was put up very recently, but we have started a project in 2002, 2003. Yes, I think people can ask questions. There is only 10 minutes, not even 10 minutes.
Sir, there is a query from Dilip Kumar, sir. Please.
Yes, please. Please ask.
Sir, I want to know name of the common algal species we can use wastewater
Okay,
treatment.
that is a very good question. In fact, I expected this. Actually, what we do is wherever we go to the industry, we try to collect algae from there itself.
Yes,
Mostly from the wastewater that is there, and that is already acclimatized to the local conditions. In that case, we bring them to the laboratory and then grow them in large scale and take them back and then put them back into the system so that the local algae will start growing. You're not introducing anything new. Very rarely we introduce algae from some other place. So algae like chloral, Senadesmus, Chlorococcum, and Desmococcus, Desmococcopsis, Oocystus. These are all species I think that people, I mean, we have been using in various species places. And in fact, the first project we did use Coccus in the first project, and then some places we also used Gonium in UK and in Colombia. Yes.
okay. No other questions from participants.
Probably I've been very fast. They are not able to follow.
No, sir, you had delivered input.
Because I delivered only 15% of what I prepare. I mean, that normally takes. I mean, it's all 45 minutes. Dr. Sajini is already there. He's waiting. I want to listen to him. In fact, Dr. Sajini, how are you?
Good morning, sir.
Good morning. I'm waiting for some questions. There is anything 4 more minutes is there. I mean, I think they can ask some questions they want.
Okay. If any participants have any query, please ask in chat box. Okay.
You can share my email address to people and they can contact me. Next time when I come to Rajasthan, I will visit definitely. I was there last month.
Oh,
I was traveling Kota and then I went to Jaipur. I was there for 3 or 4 days in January, 20th, 21st, and all that. I regularly visit there, and I know people from Jaipur University, a lot of people.
thank you so much, sir.
Thank you.
Now, I would like to invite Organizing Secretary of the program, Dr. Dilip Kumar Rathod, sir, to deliver vote of thanks to Dr. Siva Subramaniam. Dr. Dilip Kumar Rathod, sir.
Thank you, ma'am. Sir, you came near our city because Bundi far away, only 40 kilometers from Kota city. And
Next time I will come definitely. I will not. I mean,
most
I will not. I know because I like Rajasthan always. The food is very good. The people are very nice. In fact.
thank you very much, sir, and welcome again. And now I going to give to vote of thanks to sir. It is our great pleasure that we have with us international renowned personality related to algae, Dr. Siva Subramaniam Velisamy, sir, who honored us by giving his precious time. Dr. Swami Velisamy, sir, is a well-known algologist and he accepted our invitation at the very first conversation. I, on the behalf of Government College Bundi and whole organizing committee, thank you very much, sir, for, from the bottom of my heart. Sir, we learned about Ficus technology from algal growth and removal of cyanobacterial sheath. We come to know about many projects, both national and international level, and use of algal technology for projects like Clean Ganga Mission and Tata Steel Jamshedpur Lake Restoration in Jhansi, Mosam River Restoration, etc. And so once again, thank you very much, sir, and thanks again. Now I invite to Dr. Nitika Singh to welcome and introduce our next renowned speaker, Dr. Tajuddin sir, who is well-known algologist, Dr. Nitika madam.
Thank you so much, sir. Now, I will take the opportunity to introduce our second eminent speaker of today, Dr. N. Tajuddin, sir. The words fall short in front of his vast experience. Currently, he is working as professor, Department of Microbiology, School of Life Sciences, Bharti Dasan University, Tiruchirappalli, Tamil Nadu. Apart from that, Sir is also working as principal investigator of a project sponsored by DBT entitled National Repository for Freshwater Microalgae and Cyanobacteria. Uh, Sir is also serving as program coordinator, MSc Life Sciences, coordinator, DST PURSE program, convener, world class curriculum development cell in biological sciences, editor in chief, Bharti Darshan Journal of Science and Technology. He has also served as syndicate member, senate member, dean, faculty of science and engineering and technology, head department of microbiology, and president alumni association. He has published 337 research, including 3 papers in scientific reports and journals of antibiotics, Nature Group, 27 book chapters and review articles, a book on microbiology and immunology at postgraduate level in Tamil, lab manuals, and 6 edited books in CRC Press, Elsevier, and Intech publishers. He has received major research projects from the government funding agencies, namely DBT, DST, UGC, and MOES, of approximately 5.27 crores. Dr. Tajuddin guided 33 PhD candidates, 14 postdoc fellows, acted as doctoral committee member of 166 PhD candidates, and currently guiding 7 doctoral candidates on various aspects of microbiology. Sir has received many prestigious awards, including Dr. G.S. Venkataraman Memorial Best Scientist Award, Dr. M.S. Swaminathan, NABS Leadership Award from National Academy of Biological Sciences, TA and CA Award from Government of Tamil Nadu, K. Chidambaram Memorial Annual Award 2019 from Fisheries Technocrat Forum, Chennai. Today, he will be delivering his talk on a very interesting topic, microalgal identification, conventional and molecular methods. I welcome you, sir. You can start your presentation. Dr. N. Tajuddin. Sir, you can start your presentation.
I'm sorry for the internet disturbance, sir. Is it audible to me, to you?
Yes, sir.
So is my voice is audible to you?
Yes, sir.
Okay, I'm going to share my slides now. So, at the outset, I thank the organizers for giving this opportunity, and also I am very happy that Dr. Dr. Subramanian, who is a world-renowned phycoremediation technologist, gave a wonderful lecture after his vibrant talk. Now I am going to talk on very basic aspects of microalgal identification, conventional and molecular taxonomic methods. But these techniques are really needed. The of the organism. We cannot do any molecular biological works. Even for patenting, we need the authenticity of the identification.
Sir, please unmute your mic. Sir, please unmute your mic.
Is it clear now?
Sir, now you are audible. Sir, there is some connectivity issue. Can you please stop your video? Sir, your voice is not clear, sir. If you stop your Sir, if you stop your video, then maybe your voice would be more clear. So there is some connectivity issue, I think. Your voice is not clear.
My voice is audible now?
Yes, sir. It is audible now.
Okay. Shall I proceed?
Yes, sir.
Okay. No, 45 minutes. Okay. So I like to share the experience I got it on microalgal taxonomy method. Initially, the morphology-based technology identification is very badly needed. So coming to the taxonomical aspects, taxonomy is an ever-changing, controversial and exciting field of biology because in decades to decades, so many taxonomists are there. They identified the species on their own view of identification system. That's why one taxonomy identified one organism. After a few years, the same organism identified the other scientists, other taxonomists in different way. So for this, we need the extensive data on the why we are identifying this organism based on some morphological properties. And the taxonomy necessitates extensive knowledge of the techniques of data acquisition and very latest in field condition. The morphology is different. Some of the cyanobacteria, if I say, for example, nostoc and microcolia species in field condition, the nostoc is looks like a mucilaginous matrix or ball. Based on this mucilaginous assemblage, we call it as a nostoc. Whereas if you are transferring the nostoc colonies and by repeated subculturing in the laboratory condition, the nostoc filaments will not produce the mucilage, because in the natural condition, to avoid the stress during the drought period, the organism liberating the mucilage and it imbibes the water for a longer period for their survival. Whereas the same organism, if we transfer to the culture medium, so throughout the year, it is exposed to water. So because of the presence of water, the organism will not liberate the mucilaginous sheath. So that's why if there is no mucilaginous sheath, the nostoc is named as anabina. So anabina, there will not be any mucilaginous sheath. Such type of misleadings are there. For this, we need some extensive knowledge, not only the morphological background, but also the biochemical and molecular techniques should be involved to segregate and identify exactly the species. Nowadays, the most important point is the taxonomists have now become endangered species category, because all the students, upcoming students, they want to do the biochemistry, molecular biology, genetic engineering, and biotechnology, and so on. No students are interested on doing the taxonomical work. But I'm also during my research scholar level stage. I'm not interested in taxonomy. But my guide, Dr. G. Subramaniam, he insisted me to do only the taxonomy, and I spent my whole 3 years on taxonomy of microalgae and biodiversity identification and so on. Because of the taxonomical work, I am very proud to say that based on my PhD work, we developed one national facility for marine cyanobacterial center in Bharathasan University, because during my work, I identified and isolated, purify and maintained the germplasm of marine cyanobacteria. Later on, this marine cyanobacterial germplasm is converted as a national center. So this is again another very important aspects. We developed one national center. After that, I involved in the freshwater microalgae and cyanobacteria biodiversity, taxonomy and identification. And recently we established the second national repository, that is National Repository for Freshwater Microalgae and Cyanobacteria. The two centers based on the taxonomic aspects we developed in Bharathasan University. So if you see this microalgal and cyanobacterial biodiversity, here I like to mention that the cyanobacteria previously called it as a blue-green algae. It comes under the algal classification system. But because of the prokaryotic nature, this so-called blue-green algae is transferred to the bacterial classification system and called it as a cyanobacteria. So here, whatever I mentioned as a microalgae, it includes the cyanobacteria also. So we need not go for any special environment to collect the microalgal and cyanobacterial biodiversity, because wherever sunlight and moisture is there, definitely you can get this growth and survival of these organisms. That's why this organism is respected as cosmopolitan in distribution and primary producers. So imagine, without primary producers, there will not be any primary consumers or secondary consumers or tertiary consumers like larger fishes. So I like to say that, so if there is no phytoplankton, that includes microalgae and cyanobacteria, there will not be fishes. There will not be any fishery community. Definitely there will not be any fishery ministry. The tiny microorganism reflects up to the fishery ministry level. So now almost all the countries, globally the scientists agreed that the microalgae and cyanobacteria is only the sustainable bioresource for fulfill to satisfy our future requirements. It is agreed by all the scientists globally. And if you see this slide, this is one sewage water pond. It is there in our university girls' hostel. Sewage water is dumped in a small pond. You can see the bloom of algae. So if you see under the microscope, you are getting the very beautiful creatures. So it is available everywhere. And mostly all the water bodies in the cities and all the new and famous places, all are eutrophicated, so are polluted. So you can collect only the pollution tolerant algae, microalgae and cyanobacteria, not the 100% diversity. So for this, the pollution tolerant organism in the eutrophic region, it forms the extensive blooms. Whereas if you want to identify the 100% biodiversity, you have to go to the places where it is water bodies are unpolluted, undisturbed. So there only we can collect the entire biodiversity of microalgae and cyanobacteria. And if you see this, there are two major groups, as I said, eukaryotic micro and macroalgae and prokaryotic cyanobacteria. So in the important green algae, it has some special features like it is a unicellular, colonial, and photosynthetic products are starch and oil, and chlorophyll. Photosynthetic pigments like chlorophyll A and B, alpha and beta carotenes are there. And next one, xanthophysi, it is called, it is a yellow-green algae. So it has excess of xanthophyll pigments. The photosynthetic products are oil and glycosan, cell wall rich of pectins. Likewise, each and every division has its own specific characteristic features. So if you go to the literature, you can you realize that each and every division is very specific and unique characteristics. Based on these unique characteristics, there are several divisions are there. Like Chrysophysi, Bacillophysi, Dinophysi, Euglenophysi, then Rhodophysi and Phaeophysi, brown algae. Mostly the brown algae and red algae called as the seaweeds or macroalgae. These organisms are, if you want to collect this whole biodiversity of micro and macroalgae, I insist you to visit the Gulf of Mannar region, particularly the Mandapam Rameswaram region. We call it as a algal paradise. So we can collect nearly 80% of the biodiversity of microalgae, macroalgae, and cyanobacteria, everything available in this small region. And this is another very famous microalgal taxonomic groups. So there are 5 major groups in terms of the utilization. One is diatoms. Now diatoms are useful for the lipid and as well as the biodiesel production, because the storage compound in the diatoms are the lipids. And green algae, there are a few green algae are commercially exploited at the industrial, like chloral, arsenic, and so on. And other group of algae, golden brown algae, primnicophytes and eustigmatophytes, these organisms are existing in our environment, but because of the absence of the taxonomist and also absence of the culture collections in the repositories, we cannot work on this one. But these organisms are equally having the potentials with cyanobacteria and microalgae. Only the absence of the taxonomist, we could not identify the organisms from the environment. And coming to the cyanobacteria, as I said, it is all previously called it as a blue-green algae. It is really structurally diverse organism, and it is a vast diversity. So unlike the bacteria, if you want to identify the bacteria, the morphological types are only 6 or 7, like Bacillus, cocci, spirillum, and then pleomorphic, vibrio, filamentous, and so on. And we are identifying the bacteria using the biochemical and some molecular features. And whereas identification of the cyanobacteria, because of its very vast biodiversity, vast morphological diversity, if you see under the microscope, you can identify up to the species level. So only problem arises if the organisms in the natural environment, it looks different, whereas in the repeated subculturing in the laboratory condition, it is again look different. For this, we have to use the molecular tools to identify these organisms. So in cyanobacteria, there are major 5 orders are there. One order belonging to crococcyles, that is all the unicellular and colonial forms. There is no differentiation in the base and affects cells spherical or elongate reproduction by fission. The advantage of cyanobacteria is there is no mitosis, there is no meiosis, only the amitotic fission. So that's why this organism is useful in biotechnology, because the biomass production is very, very rapid. That's why, because there is no complexity in the reproduction, only the amitotic fission. The first group is mostly the unicellular and colonial forms. The second one, again, this is unicellular, but it has basal and apical differentiation, and it has the exospores. It forms the exospores, all are non-motile. This coming under the chemosiphonales. And the third category, that is plurocapsules. This is again unicellular, and it is aggregated and looks like a filamentous. It is called false filamentous form, and also we say that it is a pseudo parenchymatous thalae with prostate and erect system. But all are maintaining its individuality. Each cell in it is maintaining the individuality. So there is no differentiation of trichomes or filaments, no hormogonia, no heterocyst, no endospores in the sporangia, only exospore producing formation. So sorry, there is no exospores, only the endospores formation is takes place in the caves of plurocapsules. And coming to other major group, one is the nostochales. Usually nostochales coming under the filamentous, trichomatous filamentous form coming under oscillatory and other genera. And in addition to this, some other group which has the heterocyst, terminal heterocyst and intercalary heterocyst. So all are belonging to the nostochales. The trichomes or filaments unbranched with the false branching coming into the nostochales. And other final branch, it is a highly evolved cyanobacterium, because it has true branching, which is called heterotrichous filamentous form with the presence of heterocysts. Likewise, we can segregate the entire cyanobacterial diversity in 5. There are some specific key to all the genera. Under the genera, the maximal number of genera we can identify up to the genus level. We provide the. So is it audible? Is it audible?
Yes, sir. It is audible. Sir, please continue.
Some net disturbance.
Sir, you are not audible now.
My
Sir, please unmute your mic. Sir, please unmute your mic. Sir, please unmute your mic. Okay.
Some disturbance are there. I'm sorry for that.
Okay. Okay, sir. You are audible.
Okay, my slides are visible.
No, sir. Please share your screen.
Okay.
जो टाइम मिलता है तो सुन लेते हैं ज्ञान ले लेते हैं
All the participants are requested to please mute their mics.
So my slides are visible.
Yes, sir. It is visible.
Okay. The second group of organism is plurocapsules, chemosiphonales. You can see this. There is a basal and apical difference is there, in which you can see the endospores, exospores. All are coming under the next category. And the third, plurocapsules. You can see the unicellular forms are aggregated. It looks like a basal, prostate, and erect system like this. So these are all the second and third category. And the fourth category is the nostochales, in which the non-heterosystous cyanobacterium like Spirulina, oscillatoria, and different types of Formidium, and so Trichodermium. This is bloom-forming organism. Everything is there in this type. Here you can identify based on the morphological structures in the apical tips region of the cells. You can see the variation in the apical region, like pinnate, very pointed tips, blunt tips, and then some constrictions in these cross walls. Likewise, you can identify based on the morphological characteristics. Here you can see this. All the four slides are nothing but the Formidium tenue, because of the environmental conditions and their proportions in the pigment presentation, like phycocyanin and phycoerythrin. If it is a uniform proportion, you can get the blue-green color. If phycoerythrin dominates, you are getting the red or brown shade like this. So for this, morphological all are same Formidium tenue, but genetically and biochemically it is different. And again, this is belonging to the oscillatoriaceae member. The Lingbaea species, you can see the lamellated sheath, the simple sheath, and with the false branching, and the apical capitate end cells. Likewise, each and every species we can identify up to the species level under the microscopic observation. This is the bundle forming trichomes in a common mucilaginous sheath. Again, this is Monocoleus cathenoplast. You can see the two different strains. One is dark brownish, and another one is the brownish. But morphologically, these two are the same species. And this is another group, heterocystis cyanobacterium called nostoc. This is the fluorescent microscopic view where the heterocysts are pigmentless, so that you cannot see any fluorescence in this gap. This is the region for presence of heterocyst, and this is again the heterocyst, not a nitrogen fixing capability with this, because there is no pigments, photosynthetic pigments. In the absence of oxygen, the nitrogenase enzyme is activated and fix the atmospheric nitrogen and convert it into nitrate and ammonia, which can be utilized by the agriculture crops. This simple heterocystis bacterium. And one cyanobacterium called called Richelia intracellularis, very short cyanobacterium, but it residing endosymbiotically within the diatom species called Rhizosolinia. These are all simple heterocystous cyanobacterium, and this is Cytonema, this is false branched cyanobacterium, and this Mustiocoleus and Hepalosiphon, they are highly evolved species because it has a prostrate and erect system and with heterocysts. So these are all different morphological types based on the measurement, cell size, cell shape, and the apical region of the filament or trichome, and the presence of heterocysts, absence of heterocysts, false branching, true branching, and so on. We can identify, because the cyanobacteria, the morphological diversity is very extensive. We can identify up to the species level. And these are all some of the culture collections we have in our germplasm of National Repository, some green algae. So we have the green algal biodiversity. If you see, each and every organism is differ from each other because of the vast diversity. And this is a beautiful diatoms, estuarine diatoms, like different. These are all the very beautiful structures, ornamentation because of the silica cell wall. So this organism also we identified up to the species level by seeing under the microscope. And here I like to show the confocal microscopic view of Cyanidesmus. In confocal microscopy, its cost is nearly 1.2 crores. We purchased under a DST Purse program. These are all manipulated colors by using different filters. You can get this beautiful coloration. And this is some Aphthenocapsa species. You see the confocal view, and then this is the Crococcus turgitus. This is again the confocal view of very beautiful colorations. These all colors are manipulated. These photographs are available in our NRMCF website. And coming to the taxonomical methods, now I'm going to talk about the molecular level taxonomy. So basically, the morphological characterization, the morphological taxonomy is very, very important because it is the base for all taxonomic methods. So without morphology, it is really meaningless to go for biochemical and molecular taxonomy. First, we have to see the structure of the organism. It is helpful for the biotechnological angle. For example, if you want to isolate the DNA, so you have to see first the morphological type of the organism. Sometimes the some unicellular is very flimsy cell wall, and the Gliocaps, some Crococcus, it has a very lamellated, very thick cell wall. So if you use this for DNA isolation, your conventional treatment of lysozyme or any other treatment to break open the cell will not useful. So you have to see the morphology, and then you standardize the techniques for breaking open the cell. So if a very thick cell wall, you add the higher concentration of the lysozyme, some sonication, or combination of different techniques to break open the cell wall and get it and isolate the DNA. Otherwise, it is difficult. So morphology is the base. So without morphology, there is no other meaningful taxonomy methods are there. In biochemical, next one is the biochemical and chemotaxonomy. Yes, in bacterial system of classification, we are using the biochemistry for identifying the lacks of bacteria, because morphology is diversity of the bacteria is only 5 or 6 types, whereas we have several lakhs of bacterial identification, bacterial species reported based on the chemotaxonomical marker. Whereas in the case of microalgae and cyanobacteria, the biochemical features are not constant. If you change the media composition and condition, you cannot get the same fatty acid profile, you cannot get the same amino acid or protein profiles, because if you change the media composition and condition, you are getting differently. For example, if you grow the algae and do the chemotaxonomic work with some heat it has stress. So if you give some heat to the organism, definitely the biochemistry will be vary. It is not constant. It depends on the media composition and environmental condition. It is vary. So we cannot say that specifically these biochemical features for true identification. Coming to the molecular taxonomy, There are several tools are there. One is DNA-based composition, DNA hybridization, RFLP, RAPD, STRR, LTRR, pulse field gel electrophoresis, plasmid profiles, and 16S rRNA sequence analysis. Among the all tools, 16S rRNA is helpful for identification of the species and the genera, even strain level. Whereas other tools like DNA-based compound, RAPD, RFLP, LTRR, these are all the tools helpful to differentiate the closely related species, not for identification. So I will justify this. So in tool for molecular taxonomy, people using 16S rRNA in the case of prokaryotes and the 18S rRNA in the case of eukaryotes. The ribosomes of 70S ribosomes in the prokaryotes and the 80S ribosomes in prokaryotes. 70S ribosomes in prokaryotes, they have different RNA molecules like 23S and 23S and 28S in the case of eukaryotes. But this only in the case of prokaryotes like bacteria and cyanobacteria, we use only 16S rRNA, and in the case of eukaryotic microalgae or macroalgae, we should use the 18S rRNA because some positive points to justify this. Okay. So as I said, the chemotaxonomical markers, the fatty acid fingerprinting, polyamines, biochemical features, protein isozyme patterns, all are substrate dependent. If you change the substrate or if you change the media composition and condition, you cannot get the same result. It differs. For example, Gyan in 1972, he screened 66 strains, and Anabino and Galactrix both are morphologically different, but it has the same profile. Likewise, sometimes different profiles will be there. And coming to phycobilly protein, again, this is the, if you change the substrate, you cannot get the same proportion of the pigments in this species. And coming to other tools, molecular tools like DNA, base composition, restriction, I'm going to explain little more about in the subsequent slides. If you see this slide, the DNA-based composition of various organisms, prokaryotes, eukaryotes, animals, protists. If you see this slide, there is no uniqueness in the DNA composition. All are overlapping with each other. So we cannot say based on this GC percentage, this is in this group of organisms. Like because this slides clearly indicated that there is no uniqueness of GC percentage to any specific organism. All are overlapping range with each other. And even in the cyanobacteria, for example, if you see this slide, the Xenococcus species, it is unicellular and aggregated colonies. And the second one is Orthospira. It is the trichomatous spirally twisted colony, trichome. And Cytonema, it is a heterocystous false branched form. All are morphologically totally different, but they have the same GC profile. So that's why the GC percentage is not really useful for identification of the organism. But if you want to develop any molecular data bank, you can use these characteristic features and develop the data bank for each and every isolate that you have in your germplasm collections. And this is some basic information about the ribosomes in 70 prokaryotes. There is a 70S ribosome which has 2 subunits in which 23S rRNA and 5S rRNA, and a small subunit has 16S rRNA. Whereas in the case of eukaryotes, it is 80S ribosomes. There are 2 subunits in which 28S, 5.8S, and 5S, 3 different RNAs in the largest subunit, and 18S rRNA in smaller subunit. So now, why we are specifically using the 16S rRNA for molecular identification of prokaryotes? Because it has the regions of nearly universal sequence conservation. If you analyze different types of prokaryotes, in particular region of the DNA, it is conserved for genus level. In particular region conserved for species level. In between, some variable regions are there. So it reflects up to the strain level differentiation. And the length of the 16S RNA is around 1500 to 1600. It has it is very easily aligned. When you are making the phylogenetic tree, the alignment is easy alignment is possible, and it is very large informative. Whereas in the case of 23S RNA, it is very large molecule, it not having sufficient amount of the conservation, and also is statistically very difficult. Whereas in 5S rRNA, it has only 125 nucleotide length. The amount of information we are getting, which is not sufficient for the molecular taxonomy as well as making the phylogenetic tree. For this, the 16S rRNA is a very ideal molecule. The length is very convenient, and it has non-conserved regions also, particularly variable and hypervariable. So if you have the same species of nostoc or Formedium tenue, which is isolated from different environment, different locations, and morphologically all are same. But if you are going for the 16S rRNA, so some regions are different. So the genetic makeup of the each and every isolate is different from each other. But there is a specific conserved and non-conserved regions which fits the genus level and the species level identification. And in addition, largest database is available. For example, in ribosomal database project, there are nearly thousands of sequences already deposited. If you have your new sequences, just to go to the this database and submit your sequence, within few minutes, you get the results. Your sequence is 100% matching with this particular species, 99% matching with particular species. Likewise, you are getting the data in no time. And this is the secondary structure. For example, in the case of E. coli, there is a darker region and red region, orange region, and yellow regions are there. The darker regions are highly conserved regions. We can use it for the identification, and variable region is useful for the strain level variation. And these are all the methods we have to follow for DNA fingerprinting, particularly the molecular taxonomy, DNA isolation, PCR amplification, cloning, transformation, plasmid isolation, gene sequence, and sequence analysis. These are all the various steps are there. So you may ask question that I'm talking about the 16S rRNA, but you are talking here, you are mentioning the DNA isolation, how it is relevant. So because, you know, we do isolate the rRNA from the ribosomes, and then and you know, half-life period of the rRNA is very less, and it is very difficult to maintain the RNase-free environment, so easily degraded. And even if you are isolating the rRNA, you go to the reverse transcriptase PCR and then make the rRNA molecule into DNA molecule and go for the again normal PCR to amplify. So finally, we are getting the DNA sequences also, DNA 16S RNA. For this, to eliminate all the complex processes. So DNA is a sturdy molecule. Here we are using the 16S rRNA coding regions in the DNA by selecting the specific primers. And we can easily amplify or take the Xerox copy in lakhs within few hours. And the PCR, second one is the PCR amplification. Once you isolate the DNA, use the specific primer and amplify the 16S RNA genes. So if you want only the partial sequences, you just after amplification, you send the product to sequencing companies, you are getting the partial sequencing of nearly 800 to 900 sequences. Whereas if you want all the 1600 sequences for your purpose, so you have to clone your PCR product with the cloning vector, that is called ligation or cloning. And after the recombinant molecule plasmid is there, then you transform this cloned recombinant molecule with the 16S rRNA product to the E. coli cells. That process is called a transformation. And after this, you multiply the transformed cells overnight, and you are getting the millions of cells, and you isolate the plasmids, and from this plasmid you go for the DNA sequencing technique, and once you get the sequencing, you go for the sequence analysis and make the phylogenetic tree. All the comparison, statistical comparison, everything is possible once you have the sequence, and you can submit the sequence in the gene bank also. And initially for molecular taxonomic work, we initially selected the cycas plant. In cycas plant, it has this special corollary roots. If you're taking the cross section, there is a algal zones is there, cyanobacterial zone is there. So far, only one in literature, Nostoc cycadea is only reported, only species reported as Nostoc cycadea. Whereas in our morphological and molecular fingerprinting technique, we isolated more than 19 different strains of cyanobacteria in this zone, and one new genera like Calothrix also reported in this one. And we did the molecular phylogenetic aspects. We published this paper in very good impact factor journal from USA. And another symbiosis we selected is lichens. In lichens, some cyanobionts are there. It is called cyanolichens. We isolated the cyanobionts from the lichens, and we did the molecular phylogeny. And these are all the isolates from the lichens. And another symbiotically associated or cyanobacterium is in azolla. So it is, the pteridophyte in the cavities. Usually, Anabina azole, only one species so far reported, but from our metagenomic study, we reported that more than 200 different strains of cyanobacteria residing in this azolla cavity. In addition to this, nearly 500 bacterial symbionts are there using our metagenomic study. So if you are using the conventional isolation method or purification method, we can isolate only less than 10 different strains of cyanobacteria and only less than 50 different colonies of bacteria. Whereas if you are going for metagenomic approach using the 16S rRNA, we can report more than 500 or 600 endosymbionts in this azolla species. So initially we collect the DNA, whole genomic DNA from the azolla. In the azolla genomic DNA, the one DNA from the azolla, eukaryotic azolla DNA, and other two different DNA, one is from cyanobacterial symbiotic 16S rRNA as well as bacterial symbiotic 16S rRNA. So we use the crude extract of all the symbiotic forms and isolate the DNA. So we have a crude extract of cycas, corollae roots, and then azolla and lichens, and we have the DNA of each and three different sources. And this is the basic DNA isolation method. I am not going to detail about this. Only three major steps are involved in this DNA isolation. One is the cell break open the cells using the glass powder, SDS, and phenol chloroform. And the second major step is the extraction using the phenol chloroform. And finally, the precipitation by ethanol, 100% ethanol with sodium acetate, and then finally store it in TSD buffer. This is the basic three steps involved in almost all DNA isolation protocols. And once you isolate the DNA, you confirm the presence of DNA using the agarose gel electrophoresis. Since it is a high molecular weight protein, high molecular weight DNA, it moves only very near to the well where we loaded. If it is a lower molecular weight, it moves far away from the wells. So you confirm the DNA, isolated DNA by using the agarose gel electrophoresis. There are three different techniques are used for resolving the DNA. One is acrylamide. This is the sequencing gel. Very less molecular weight, we can DNA. We can use this gel. And the agarose gel electrophoresis. This is used for our genomic DNA visualization. And another one, very high molecular weight DNA, we have to use the pulse field gel electrophoresis techniques. So there are three techniques are available. And once you isolate the DNA, go for the DNA amplification by using the PCR reaction. So here, this is the PCR component. It has the two primers, that is forward and reverse primers. So we use the universal 16S rRNA primers. So it decides where to start and where to stop in the 16S rRNA region of the coding region of the genomic DNA. And other ingredients like DNTPs, tag DNA polymerase, and buffer, then DNTP mix. So all are required for this standardized protocol. And temperature cycle, denaturation, primer annealing, polymerization, different temperatures are there. In PCR, the denaturation and the primer extension, that is polymerization temperature is unique to all the PCR reaction. Only difference in the primer annealing. This depends on the length of the primer, length of the nucleotides in the primer sequence. Based on the length, there is one calculation. So we we can decide what is the optimal temperature and optimal duration for the primer annealing, so that it is available. For example, consider this is the genomic DNA. So we have a primer which fits only the complementary region of the 16S rRNA coding region in the 5' end as well as the 3' end in the opposite strand. On PCR reaction, so this is just the explanation how PCR occurs. This is the initially double stranded DNA, and 94° the double stranded DNA is denatured, so break open all the hydrogen bonds between these, and then primer annealing in the 50°, for example, it go and bind exactly in the 16S rRNA coding region of the both strands, and then it the tag polymerase binds, and then it extension takes place. Now we are getting in the third cycle extend 72°, we are getting the two strands, but it is having the unequal length. And in the second cycle, again denaturation, and then primer annealing, and then extension is there, and you are getting 4 different fragments. In the third cycle, again the denaturation and the primer annealing, then the extension takes place. Here you can see this these two more strands are exactly same length. So this is in the third cycle. In subsequent cycles in PCR, we are running for nearly 25 to 30 cycles. So during the course of varied temperature exposure, the overhanging regions are degraded. So finally, we are getting the exactly same length of 16S DNA molecules. So this is how, what is the proportionate amplification of this one. Initially, the first cycle only 2 strands are there. In 6th cycle 64 strands, whereas in 25th cycle we are getting 83, more than 83 lakhs of 16S rRNA amplified products in a small 0.5 ml F&F tube. So this is very really amazing technology discovered by Kali Mills. Once you isolate the DNA, amplify the DNA, you go use the agar-agar electrophoresis, and then you visualize your presence of 16S rRNA products. So we have to use the positive control and negative control. Positive control is useful for checking the purity of the PCR kit we are using, activity of the PCR kit, because in past PCR kit they are giving the template DNA and as well as the primer DNA. So if it you are getting the band, that means your PCR kit is very active. There is no degradation in this. The negative control is equally important, because in this PCR mixture we are not adding any template DNA. Instead of template DNA, we are adding all the ingredients. But in this negative control, there should not be any band. If it is a band is there, definitely your PCR kit is contaminated. So likewise, we have to run with the positive and negative controls. These are our samples. And once you have the PCR product, you ligate with the PGMT vector or any PCR1 vector. There are so many commercially available vectors are there. So you have to ligate or clone your PCR with this vector using the DNA ligase enzyme. And to check this, whether you have the recombinant molecule, that is the vector with the PCR insert, you again run the gel. So this is the vector DNA, and this is somewhat higher molecular weight DNA, because it is a vector plus the PCR product. That's why it receives the higher molecular weight. So from this gel, we confirm that we have the cloned product, that is vector with the PCR insert. So once you confirmed, you go for the transformation technique, and this is some details about the vector. So the vector is PCR 2.1, in which there is a lag site, that is beta galactosidase enzyme site is there. So in which we are getting the linear molecule where there is a disturbance of the vector. So it is initially linear, and the vector molecule in both 3' end, it has a T overhangs. So whereas in PCR product we developed the another specialty of the tag polymerase enzyme, it has the template independent addition of one adenine molecule in its 3' end. So as a result, our all the PCR fragments, 16S RNA genes having the A overhangs. So if we mix the vector with T overhangs and A overhangs in the PCR product, it is easily cloned, because it is complementary. that is called directional cloning method. So we direct both the molecules to clone exactly with this. That's why in the PCR product, the cloned product, if it is a recirculated plasmid, the lag Z site is intact, whereas in the recombinant plasmid, it is the lag Z site is disturbed with the insertion of the PCR product. So next step is, once you confirm that you have the recombinant molecule, that is vector with the PCR insert, and you transform the recombinant plasmid to JM109 competent cells, which are very defined cells. Originally, it is ampicillin sensitive, and all the characteristics are well known. And you mix it and then incubate for 20 minutes, and then you just pour or load or spread plate the competent cells which are transformed with the recombinant molecule, and you spread on the plate which containing the ampicillin and the XCOL and IPTG. These are all the chromogenic substrates. So based on this substrate, if there is a recirculated plasmid is there, it reacts with IGAL and XGAL and IPTG. It forms a blue colored colonies. So as a result, we are getting the blue-white colonies in the after transformation. For example, you can see this blue-white colonies. So blue colonies are just a recirculation of the vector. There is the lag Z site is not disturbed. So because of the beta galactosidase enzyme, it reacts with the XGAL and IPTG, it forms the blue colored colony. Whereas in all the white colored cells, which denotes that the plasmid is not intact, the lag Z site is disturbed with the insertion of the PCR product. So the lag Z enzyme is inactivated, and we are getting the white color colonies. The untransformed cells, because it is ampicillin sensitive, it won't grow in this medium because it is containing the ampicillin. So from this plate, we transform each and every clone to a defined number. In the other plate, this is called the library, cloned library, and we can number it 1, 2, 3 like this. Each plate up to 50 clones we can incorporate, and we can load it. So this is the clones. And from this clones, we select each and every clone and isolate the plasmid, and then you sequence the plasmid, and finally you are getting the 16S RNA sequences. And after getting the blue-white colony, you select the white colony and then isolate the plasmids. The DNA method is somewhat relevant to the DNA, genomic DNA, with some variations, small variations. And as I said, initially we use the crude DNA extracts of the Azula or Cycas plant, and we are using the universal prokaryotic 16S primers. So it amplifies the bacterial 16S rRNA and cyanobacterial 16S RNA from cyanobacteria. So to identify which of the clone is coming under cyanobacterial 16S and which of the clone coming under the bacterial 16S, by using the dot blot hybridization technique using the cyanoprobe. Cyanoprobe means it is a small oligonucleotide. It is kind of highly conserved for the cyanobacteria, which is tagged with some chemical dyes. So we use the cyanoprobe, and this is the dot blotter apparatus in which we are placing the zeta probe membrane, and then we load from the top and each and every clone, isolated plasmid on the top, and finally, if you are hybridizing with the cyanoprobe, only the cyanobacterial 16S rRNA or that plasmids are retaining the blue color, and whereas some of the plasmids which are bacterial clones, it is not taking the cyanobacterial probe. So it is we are getting nothing. So if the blue clones are denoted as the cyanobacteria, the other empty region, empty dots, it is called, it is considered as the bacterial 16S RNA. By this way, we can differentiate both. And after confirming this, if you want to select the cyanobacterial plasmid, go for the sequencing. This is, for example, this is the vector DNA, and this is our PCR product. We are again using the PCR thermocycler for gene sequencing, that is called Sanger's dideoxy chain termination method we are using. And in the DNA sequencing, we use some sequencing primers, these are the sequence binding sites, and we use the forward and reverse primer as well as in a normal PCR, and it's get amplified. Here you are using two different nucleotides. One is a deoxynucleotide and another one is dideoxynucleotides. Wherever the dideoxynucleotide binds with the complementary nucleotide in the strand, there is a chain termination occurs. So this is feature is helpful for the DNA sequencing, and this all the different DNTPs like reoxynucleotide and deoxynucleotide, ribonucleotide, and dideoxynucleotides. If you see this molecular structure in ribonucleotide, there is a 2 OH group, and deoxynucleotide 1 OH group, and dideoxynucleotide there is no OH group. So this if the dideoxynucleotide binds with the complement region, there will not be any extension of the DNA, but it terminates. So likewise, for gene sequencing, we have to make 4 different reactions, one tube containing all the ingredients like DNA template, that is a recombinant plasmid, 16S rRNA plasmid, and then other DNTPs. In addition to this, dideoxynucleotide also, and then buffered, all the other DNA polymerase, everything is included in 4 different reaction tubes. And if you see this slide, how the sequencing termination occurs. For example, this is the 16S rRNA template DNA. So you are using the sequencing primer. It go and bind with the complementary region of the 16S rRNA DNA RNA, and then we use the tag polymerase enzyme. It adds, if it adds the deoxynucleotide A, there's an extension takes place. But if you, instead of dideoxynucleotide binds with the complement region, there is a chain termination. So we are getting some different molecular size fragment. Likewise, in next cycle, wherever the dideoxynucleotide binds with the thymine molecule, thymine ATC, TTC, there is a termination. Likewise, it is randomly. Okay. So wherever T is there, if it is a deoxynucleotide, there is a chain extension takes place. But dideoxynucleotide binds with the particular region, there is a chain termination. Likewise, you are getting different molecular weight fragments. So this is in the case of the dideoxy A tube, whereas dideoxy T, dideoxy G, dideoxy C, different, all the other 3 different reaction mixtures, we are getting the same kind of different molecular weight fragments. So this different molecular weight fragments can be resolved in bisacrylamide urea gel electrophoresis. This is one manual sequencing. I did it in my US laboratory under my overseas associates award. Here, the first lane is A, second lane is T, and G and C. Likewise, you load it, and then you can see after running the gel, all the DNA fragments, different molecular DNA weight fragments are highly resolved. For example, you can see this. This is from 3' to 5', base to top. You can read it, and this is the actual 16S RNA sequencing. This is, we are using the S35 autoradiograph and exposed to the X-ray film and develop the X-ray film. Then only we can get this kind of results. And we can read from the base to top. And this is the now automated sequencer. Here, instead of 4 different reaction mixtures, only a single reaction mixture in which all the dideoxynucleotides with a different tag of color were incorporated. And based on the molecular weight differences, the lower molecular weight reaches to the base. Because of the lower molecular weight, in the basal region, there is a fluorometer, that is a detector is there. The laser beam, it detects the dideoxy molecule which is attached with some fluorochrome pigment. If it is a reddish one, you can get one single red, that is A. Then the other second nucleotide comes, terminated chain with the G, dideoxy G reaches to the detector, and again this detector, and you are getting the other peak. Likewise, based on the molecular weight, if you are running the gel one by one, the laser detector detects, and based on the coloration, we are getting the different peaks. So this is the actual sequencing method it is more advantageous because we need not spend money and time and even radioactive molecules for usage in the case of manual sequencing. It is avoided. Here, very simple and a single reaction mixture, you can easily amplify or and sequence the 16S RNA molecules. And once you have the 16S RNA molecule, you go to the RDP database and just cut and paste your sequence. This is the partial sequencing, and it will tell our sequences are exactly matching with the nostoc muscorum in particular region. This particular region, our sequence is exactly matching. So this is the primary results we are getting. So once we are getting the 16S RNA sequences, we have to deposit our sequences in the gen bank. There are 3 different gen banks are there. One is NCBI, then another one is EMBL, another one is DNA database of Japan. So if you deposit your sequence in one, so automatically your same sequences with the same access number, you can get it from the other database also. And if you submit the sequence, there are 2 options in the submission. One is Bankit and another one is sequence. In Bankit option, if you have 1 or 2 sequences to be submitted, use this one instantly and cut and paste it, and then you can get the access number. If you have hundreds of sequences, you go to the sequence format, download it, and literally load all the 100 or 200 or 500 sequences, and then you can submit at a time, and then you can get the access number for all these. And if you go to the Bankit option, it asks only 4 questions. One is the general information, and another reference information, that is source information, and the DNA source. The general information is what is the type of organism you used, and whether it is a prokaryote or eukaryote, you have to mention. And reference information, whether it is a 16S RNA or 18S or any introns or any ITS region, you have to give it. And reference information is your data, your name, address, who are the persons involved. And source information is what is the type of rRNA used. And there is a box, and you just cut and paste your sequenced rRNA molecule and submit it. Immediately you are getting the access number, that is Bankit access number. They will take at least 3 or 4 days. And finally you are getting the access numbers. So this is one sequence we deposited in the GenBank, that is NCBI. You can see our, this is the page we are getting for our sequence deposited. And this is from EMBL database, and another one is the DNA database. Our sequences are there. And once you have the sequences and you have to make the phylogenetic tree, there are several methods, and phylogenetic packages are freely available. You can use it. And usually we are using the Mega and Phylip software for making the phylogenetic analysis. And after making this software, so we are getting the phylogenetic tree. So this is 3 are from lichens. So because in India, for the first time we isolate the cyanobionts and made the sequences. So because of this, this kind of sequences are not so far deposited in any part of the world in the gene bank. This is the unique one. That's why our sequences are very unique, not matching with any of the branch. So it is separate. And another one is Cycas, symbiotic cyanobacterium in the coralloid roots. So here we reported one Calothrix species also. It is very related to Calothrix brevissima. And some nostoc species related to some nostoc linkia. Another one is some nostoc muscorum. Like this, we are getting the phylogenetic relationship. And this is from Azolla. So Azolla we are using. We selected 5 different clones. So all the 5 different clones are heterocystus cyanobacterium. That's why it is fixed. It is fixed in the heterocystus branch. So that's why this is the kind of analysis we can make. Our sequences compare and contrast to make the phylogenetic relationship with the other species. And this is some bacterial bank. Since we have the metagenomic approach, there are nearly 500 different clones of bacteria. We got it from the Azolla whole genomic DNA. We selected randomly 5 clones and sequenced it. Here, all the 5 clones are not close with each other, all are very distantly related with each other. So that's why we can say that we know only less than 20% of the bacterial diversity we are familiar with this, because most of the bacteria we cannot grow in the culture media. So we don't know the actual culture requirement for the growth and development of the all the 100% microbes existing in the environment. So that's why here you can see among the 500 clones, 3 clones are not same. All are totally different. And other methods like RAPD fingerprinting also. Here the RAPD fingerprinting is useful for differentiating the close related species, not for the mere identification. Here the primer used is not meant for any gene. So it is very short nucleotides. Based on the very small nucleotide sequences, it randomly bind with the complementary region, and we are getting the banding after using the PCR. For example, this is our template DNA. Our very short DNA is bind with randomly with different parts of the genomic DNA, and there are some rules are there. Here, it is very far away from this. There will not be any amplification occurs, and it is the same direction. Again, there is no amplification of, and we are not getting any bands in this region, and this is very away from this. Again, we are not getting any amplified product, and you can see this this two particular regions, it is somewhat closer, and length is very convenient, so we are getting the banding pattern. So this is the basic for the RAPD technique. Here the primer is not meant for any genes. It is randomly bind based on the very small length, like 5 to 7 nucleotide long. And based on the RAPD fingerprinting, we use the cyanobacterium isolated from the cycus. There are nearly more than 25 different isolates of nostoc and new calotherapeutic species we isolated. And we make the RAPD analysis. Here you can see all the 18 isolates are not matching with each other. So it has the very high genetic variation in the among the isolates. That's why even though this morphologically same, but the genetic makeup will be different. And based on the banding pattern, we measure the molecular weight of each and every band, and we can make the phylogenetic tree. Here, even though some of the clones having the 14 bands, but the same 14 number of bands, but if you see the molecular weight, it is entirely different. So likewise, we can differentiate this, and based on these differences, we make the RAPD fingerprinting, we make the phylogeny of our isolates also. And this is another work I like to mention here. We selected the Synagogus elongatus, same species, but isolated from different geological locations of India, one in Andaman Islands, one in Lakshadweep, one in Kursul Island, one in Kanyakumari, one in Goa, like this. We have different isolates of same species, and we make the STR, that is short, randomly repeated, repetitive PCR, and we are getting again the unique banding pattern is not matching with each other. From this, we can say that the species are entirely different from each other. And there are some softwares are freely available. This is GeneB structure prediction software. It is freely available. If you go to the software, if you cut and paste your 16S rRNA or any kind of sequencing based on the complementary region and uncomplimentary region, we are getting the different stem and loop structures. So by this way, instead of looking the sequences, by seeing the secondary structure, we can say that these sequences are different from each other. So even though the same species, for example, nostoc and this is nostoc, they are based on the branching pattern, we can say that it is different. And another software freely available is NepCutter software from New England Biolabs company. Instead of using the RFLP, that is using the restriction enzyme to get the different banding pattern, this is the in silico method. So if you insert our sequence, so this is our 16S RNA gene sequences, insert the sequences, this sequence will be matched with so many restriction enzyme available in the data bank, and it matches. And here you can see the most of the restriction enzymes residing in the terminal region of the 16S rRNA. Whereas in some other non-stock species, here the restriction enzymes are randomly distributed throughout the 16S rRNA. By seeing this presence of restriction enzymes sites, we can say that it is different from each other. So this is another way to resolve the closely related species also. And as an academic point of view, we made the protein fingerprinting by using the SDS electrophoresis. But as I said, it is not unique. If you change the condition, you are not getting the same protein profile. But for academic curiosity, we make the SDS phage electrophoresis of all the 25 isolates, and you can get the different banding pattern based on the RF value of the each and every banding or molecular weight. We can make the phylogenetic tree based on the protein, but it is not constant. But it is not advisable, because some of the species are related with the unrelated species also. For example, the oscillatoria, it is a filamentous form, non-heterocystous, and cytonema is a heterocystous form. So it is not really closely related, but if the protein fingerprinting, you are getting this kind of phylogenetic report, but it's not useful for making the identification or phylogenetic analysis. And we made some M13 fingerprinting of marine cyanobacterial strains, and we published this paper in Journal of Applied Phycology. We are getting the very unique banding pattern of each and every organism. So as I said, these results will be used only for differentiating the very closely or distantly related species. And coming to the final part, barcoding. Barcoding is really need of the hour to identify, and in biotechnological aspects, we need the barcode. So DNA barcode is nothing but a short DNA sequence. That is very important, and from a uniform locality in the genome. This is most important point, and used for identifying the species. So that's why we can use this barcode. And Paul Hilbert from the University of Gulf, Canada, he proposed the barcoding of animal using the mitochondrial cytochrome C oxidase subunit, that is COI. This is very unique to the animals. So he made the barcode for the first time in plants. This COI region is not really useful, but because there's no much differences, variations in this, instead of using the chloroplast genome for barcoding. And why the COI gene, mitochondrial genes are very, very important means you can see the greater differences from the genomic DNA. For the example, the nuclear genomic DNA, it is only 0.5% variation is there. Whereas in the same cell, the mitochondrial DNA, it has 9% of the variations are there. And a high copy number, relatively few differences within the species, and introns in this mitochondrial DNA, where there will not be any introns. So that's why it is really undisturbed sequences. That's why we are people using the COA genes for the case of barcoding of animals. And barcode system is freely available in the from Canada. That is a bold system, that the barcode of live data systems. So if you have the sequences, you just cut and paste your sequences to the system, and in no time we are getting this kind of barcode. So initially we selected some of the trichoderma, indigenous isolates of fungi, trichoderma we used, and we get this barcode. So in addition, some other fungi, the penicillium, trichoderma, and so on, we made the barcode. So as a result of our molecular taxonomics studies, we made the 16S rRNA sequences of marine cyanobacteria, freshwater cyanobacteria, symbiotic cyanobacteria, actinomycetes, bacteria, subsurface bacteria. It is a very interesting kind of work. The sequences are really unique, because when we saw the sequences of 16S rRNA, there are continuously more than 20 A's or 20 G or 20 C's continuously in the 16S rRNA part. So this is really unique. And fungi, microalgae, microzooplankton, viruses, and group 1 introns. Totally 500. It is somewhat old slide. Now we have 594 sequences we deposited in the gene bank. Now we are trying to convert these sequences in the as the barcode. So in conclude, the morphological characterization, morphological taxonomy is very, very important. This is base. So if you have any confusion, go for biochemical and molecular characterization using the 16SR and gene sequencing for identification and other fingerprinting methods like RAPD, RFLP, REP-PCR, STRR, LTRR, and M13 fingerprinting. All are useful for differentiating the closely related species. So with this, I really thank my guide, Dr. G. Subramanian, for guiding me in this aspect. And thanks to Dr. Sandra Nairiki Bauer for staying 1 year in her laboratory to learn all the molecular taxonomic work. Dr. Upariti from Lichenology Division for identification of our lichen isolate collections, and Dr. Abu Bakr for identification and get the sample of the cycads, and Dr. Ramamurthy from Singapore, helpful for sequencing all our 16SR intron genes from his laboratory. And again, I thank the DBT and DOD. Now it is Ministry of Earth Science for their funding. Thank you one and all. So if you have any doubts, you feel free to ask. So I'm sorry for exceeding my time because it is a ocean. I'm just squeeze everything in a very short duration. Thank you. Any doubts or clarifications, you can feel free to ask.
Thank you so much, sir, for the excellent lecture. Sir, I want to ask a question. Please explain what was SOC medium. SOC medium.
Actually, it is in the protocol. So I don't remember now. It is nearly 10 years back. I did everything, and now it is there. If you send the email, I will give the composition, everything.
Okay. Thank you so much. I will.
It is in my record. So I will send the composition of the SOC medium and detailed protocol. I will send it to you.
Okay. Thank you so much, sir. Now, I would like to invite Program Officer of this program, Dr. Rohini Maheshwari ma'am, to deliver vote of thanks to Dr. N. Tajuddin. Dr. Rohini Maheshwari ma'am.
Namaste, sir. Myself, Dr. Rohini Maheshwari. I want to thank you, Dr. N. Tajuddin, sir, Professor, Department of Micro. He delivered a good lecture about microalgal identification. We have learned about just the conventional method. Now we have come to know about the molecular methods of algal identification. Sir, you mentioned tools like PCR, recombination of 16S rRNA, dot blot hybridization, DNA sequencing, DNA fingerprinting, automated sequencing. These all were very new to us. We get very much knowledge regarding the molecular identification. Thank you, sir, for sharing your experience with us. I, on the behalf of myself, my department, and my institution, thank you, sir.
Thank you very much. I particularly thank Dr. Dilip Kumar for arranging this excellent workshop. And also, I thank my role model, Professor Dr. V. Sivasubramaniam. Because of his support, we get some very good projects, and his encouragement and inspiration, not only to me, all the phycologists of our country. His contribution is really amazing. I thank Dr. Sivasubramaniam sir also in this juncture. Thank you, sir.
Thank you so much, sir. And thank you, ma'am. Now, I will take the opportunity to introduce our next eminent speaker, Dr. Rashmi Singh. Dr. Rashmi Singh is Chairman, Board of Studies, APS University, Rewa, and Professor and Head, PG Department of Botany, Government Autonomous PG College, Satna, MP, affiliated with the APS University, Rewa. Presently, she is also working as Coordinator, Bhopal Chapter, the National Academy of Sciences, India. She has immense contribution in the field of algal biology and ethnobotany. Dr. Singh took active part in various Indian academic societies. She is a life member or fellow member of Society of Life Sciences, Botanical Society of India, and Society of Environmental Sciences. 5 students got PhD degree under Dr. Singh and 4 are still engaged. More than 50 research papers were published in reputed journals and proceedings. She also attended and chaired a number of national and international conferences and symposium. Dr. Singh has got recognition award by 3 academic societies. She has completed UGC research project and organized 3 national symposium and workshop in Department of Botany, Government PG College Satna, sponsored by MPCST and National Academy of Sciences, India. She has edited different textbooks of botany in Hindi and English medium based on MP Unified BSc first year and BSc second year and BSc final year syllabus. She also edited a research book, Eco Development and Environment and Sustainable Management and Conservation of Biodiversity. She is associate editor of two peer reviewed annual research journal, National Journal of Life and Life Science Bulletin published by Society of Life Sciences. I welcome you, ma'am. Today, Dr. Singh is going to deliver her talk on general aspects of research methodology in the field of phycology. You can start your presentation, Dr. Rashmi Singh.
थैंक यू मैम। फर्स्ट ऑफ ऑल मैं सबसे पहले तो गवर्नमेंट कॉलेज बूंदी को ये बहुत ही अच्छी वर्कशॉप ऑर्गेनाइज करने के लिए धन्यवाद देना चाहूँगी और सबसे ज़्यादा धन्यवाद डॉक्टर दिलीप राठौर जी को भी कि जिन्होंने मुझे एक मंच दिया यहाँ बोलने के लिए, अपनी बात रखने के लिए। और इस वर्कशॉप में जो मैं शुरू से अभी जितने भी स्पीकर्स को सुनी हूँ उनके बहुत ही अच्छे प्रेजेंटेशंस थे। उन्हें सुनकर मुझे भी बहुत ही अच्छा लगा और बहुत सारी इंस्पिरेशन मिली है इनसे। डॉक्टर ताजुद्दीन, डॉक्टर शिवा सुब्रमण्यम को थैंक्स। नाउ आई एम स्टार्टेड माय प्रेजेंटेशन। मेरी स्क्रीन शेयर हो रही है आप लोगों को? एक्सक्यूज मी, स्क्रीन शेयर हो रही है?
Yes, ma'am.
My topic is general aspects of research methodology in the field of phycology. First of all, मुझसे सर ने, दिलीप राठौर सर ने कहा, रिक्वेस्ट किया था कि मैं बच्चों के लिए, रिसर्च स्कॉलर्स के लिए और कुछ टीचर्स के लिए थोड़ा हिंदी में बोलूं, तो मेरा टॉक में नहीं हिंदी में ही आप लोगों के सामने मैं रखना चाह रही हूँ। तो रिसर्च क्या है? रिसर्च के लिए एक हमारे दिमाग में सबसे पहले अगर एक बच्चा एमएससी कर लिया तो उसके मन में आता है कि मुझे रिसर्च करना है। तो रिसर्च के लिए उसके माइंड में क्या आना चाहिए कि रिसर्च है क्या? तो दिस सिस्टमेटिक रिगरस इन्वेस्टिगेशन ऑफ अ सिचुएशन और प्रॉब्लम इन ऑर्डर टू जनरेट न्यू नॉलेज और वैलिडेट एक्सिस्टिंग नॉलेज। मतलब ये दिमाग में हमारे कंसेप्ट क्लियर रहना चाहिए कि हमें नया नॉलेज जनरेट करना है या जो एग्जिस्टिंग नॉलेज है उसी को ही वैलिडेट करना है, उसी को और बेटर बनाना है। एंड endeavor to discover new or collate old facts etc by the scientific study of a subject or by a course of critical investigation। या तो हम उसकी साइंटिफिक स्टडी करें या कोर्स ऑफ क्रिटिकल इन्वेस्टिगेशन करें। इस तरह से हम अपने रिसर्च के बारे में सोच सकते हैं। इस पूरे प्रोसेस में ये फैकल्टी डेवलपमेंट प्रोग्राम है, तो मैं चाहती हूँ कि आपके यहाँ, क्योंकि राजस्थान गवर्नमेंट के से आप लोग एल्गी के एरिया में भी बहुत अच्छे से रिसर्च कर सकें, तो इसके लिए ऑब्जेक्टिव्स ऑफ रिसर्च हैं कि टू गेन फैमिलियरिटी विथ न्यू इनसाइट्स इनटू अ फिनोमेनन। कोई भी फिनोमेनन है उसमें न्यू इनसाइट, न्यू तरीके से उसे सोचना, देखना, समझना। टू एक्यूरेटली पोर्ट्रे द कैरेक्टरिस्टिक्स ऑफ अ पर्टिकुलर इंडिविजुअल ग्रुप और सिचुएशन। बहुत से अभी भी टैक्सोनॉमी की जहाँ तक अपन बात करें, अलगल टैक्सोनॉमी की, तो अलगल टैक्सोनॉमी में अभी तक हज़ारों स्पीशीज़ का इन्वेंशन हो चुका है, लेकिन उसके बावजूद अभी भी बहुत सी स्पीशीज़ ऐसी हैं जो जिनका हम नाम नहीं कर पाए या जो अभी हमारे सामने नहीं आई हैं। तो ये जो मेथड है, इसमें हम रिसर्च में हम उन स्पीशीज़ को जो कि हैं तो इस संसार में, लेकिन अभी हम उनके बारे में नहीं जानते हैं, उनके बारे में हम जान सकते हैं। तो एनालाइज द फ्रीक्वेंसी विथ विच समथिंग ऑकर्स और इट्स एसोसिएशन विथ समथिंग एल्स। फ्रीक्वेंसी कितनी बार आ रही है, मतलब ये एक एनालिटिकल डेटा हो सकता है कि कोई भी आप रिसर्च कर रहे हैं तो उस रिसर्च में वो कितनी बार रिपीटिशन हो रहा है, वो कितनी फ्रीक्वेंसी में आपका एनालिसिस हो रहा है, ये आप कर सकते हैं। या फिर आप उसका एसोसिएशन देख सकते हैं कि मान लीजिए एल्गी के केस में ही कि हम दो एल्गी का आपस में कंपैरिजन कर सकते हैं, उनका एसोसिएशन शो कर सकते हैं। टू एग्जामिन द हाइपोथेसिस ऑफ अ कैजुअल रिलेशनशिप बिटवीन टू वेरिएबल्स, दो वेरिएबल्स के बीच में क्या कैजुअल रिलेशनशिप हो सकती है, उसको एग्जामिन करना, ये भी हमारे रिसर्च का ऑब्जेक्टिव हो सकता है। क्राइटेरिया ऑफ गुड रिसर्च, अच्छा रिसर्च, हम रिसर्च तो बहुत हो रही है, चाहे एमपी की यूनिवर्सिटीज़ हों, राजस्थान, बिहार, हर जगह की हमारे देश में जितनी भी यूनिवर्सिटीज़ हैं उनमें रिसर्च बहुत हो रही है। लेकिन गुड रिसर्च के रूप में अगर हम देखें तो उसमें बहुत कम थीसिस ऐसी हैं जिनको हम गुड रिसर्च के बेसिस पे अच्छा मान सकते हैं। तो गुड रिसर्च क्या है? गुड रिसर्च इज सिस्टमैटिक होना चाहिए, मतलब प्रॉपरली स्टेप वाइज उसमें हमने सीक्वेंसिंग किया हो, उसको प्रॉपरली डिफाइन किया हो, तो वो सिस्टमैटिक होना चाहिए। गुड रिसर्च इज लॉजिकल, साइंस के केस में खास करके लॉजिकल अगर रिसर्च नहीं है, अगर आप क्यों का आंसर नहीं दे पा रहे हैं, तो अगर आप क्यों का आंसर नहीं दे पा रहे हैं तो आपकी रिसर्च गुड नहीं कहलाएगी। गुड रिसर्च इज एम्पेरिकल, ये जो गुड रिसर्च है उसको आपका जो कॉन्क्रीट डाटा है उससे एविडेंस उसमें होना चाहिए कि आप कोई भी रिसर्च कर रहे हैं साइंस से रिलेटेड, तो उसमें आपको एक कॉन्क्रीट डाटा देना पड़ेगा। उस कॉन्क्रीट डाटा के आधार पर आप उसकी पूरी एक्सटर्नल वैलिडिटी बता सकते हैं। और गुड रिसर्च इज रेप्लिकेबल, मतलब ये जो गुड रिसर्च है वो रेप्लिकेबल भी हो सकती है, उसको रिपीट भी किया जा सकता है, लेकिन इन सेंस कि आप ये बताने के लिए रेप्लिकेट करें कि हाँ, जो पहले प्रीवियस वर्कर ने काम किया था वो सही था और हम उसके एविडेंस में ये हम एक्सपेरिमेंट दे रहे हैं या हम ये एविडेंसेस दे रहे हैं। तो क्राइटेरिया ऑफ गुड रिसर्च ये होना चाहिए। अब जो नेक्स्ट मेरी स्लाइड है वो ये बताती है कि एल्गी में क्या स्कोप है। बहुत सारे बॉटनी में जो है बहुत सारे हमारे पास डायमेंशंस हैं जिसमें हम रिसर्च कर सकते हैं, लेकिन हम एल्गी में ही रिसर्च को क्यों लें, तो इसके कुछ पॉइंट्स हैं। एल्गी कैन बी ग्रोन इन लार्ज क्वांटिटी विद इन अ शॉर्ट पीरियड ऑफ टाइम, बहुत ही कम समय में और लार्ज क्वांटिटी में हम ग्रो कर सकते हैं। इन रिसेंट टाइम देयर इज मच इंटरेस्ट इन डाइटरी सप्लीमेंट, क्योंकि पॉपुलेशन एक्सप्लोजन है और पॉपुलेशन एक्सप्लोजन में क्या हो रहा है कि हमारे यहाँ भोजन की कमी हो रही है, तो फूड जो स्कार्सिटी ना हो उसके लिए हमको डाइटरी सप्लीमेंट्स के रूप में एल्गी पे भी जाना चाहिए, एल्गी के बारे में सोचना चाहिए, क्योंकि पूरे हमारे ग्लोब में 3/4 एरिया जो है वो वाटर से कवर्ड है, और ये जो 3/4 एरिया जो वाटर से कवर्ड है इसका कोई भी पार्ट बहुत कम, रेयर, कुछ मैरिन एल्गी हैं जिनको हम एज ए फूड इस्तेमाल करते हैं, लेकिन बहुत सारे ऐसे हैं एल्गी जिनमें बहुत ही यूज़फुल हैं, लेकिन हम उन्हें एज ए फूड यूज़ नहीं कर सकते, तो हम अपने डाइटरी सप्लीमेंट के रूप में इनको यूज़ कर सकते हैं। एल्गी can also be exploited for the commercial production of various products like dyes, fodder, bioplastics, ये इन सब पे भी हम वर्क कर सकते हैं। तो एल्गी को हम क्यों चूज़ करें अपने रिसर्च टॉपिक के रूप में, इसके लिए मैंने कुछ एल्गल इकोनॉमिक्स के बारे में यहाँ पर बात की है, जिससे आपके माइंड में एक चीज़ आ जाए कि हम एल्गी ही क्यों यूज़ करें, क्योंकि आप अपने एरिया की एल्गल फ्लोरा पर वर्क कर सकते हैं। उस एल्गल फ्लोरा को आप अपनी एनवायरनमेंट से जोड़कर शो कर सकते हैं कि हम किस तरह का काम करा सकते हैं। तो एल्गी एज फूड, क्योंकि मैंने अभी आपसे कहा जैसा कि इतना पॉपुलेशन एक्सप्लोजन है कि हमको फूड डाइवर्सिटी पे तो हमको जाना ही पड़ेगा। अगर हम फंजाई में मशरूम्स पर जा सकते हैं तो वाई नॉट एल्गी। तो हम एल्गी पे क्यों नहीं जा सकते? तो मेरा ये कहना है कि एल्गी बहुत अच्छे डाइटरी सप्लीमेंट के रूप में काम कर सकते हैं और इसमें काम भी चल रहा है। बहुत सारे एल्गी जो है डाइटरी फूड सप्लीमेंट के रूप में काम हुई हैं, जैसे मरीन एल्गी जो हैं उनको यूज़ कर रहे हैं हम, बहुत सारे मरीन एल्गीज जो हैं उनसे फूड सप्लीमेंट्स बना रहे हैं। लेकिन इनमें है क्या-क्या ये हमें पता होना है, तो हमें इनकी बायोकेमिस्ट्री पे जाना पड़ेगा कि ये रिच सोर्स ऑफ प्रोटीन्स हैं, फैट्स हैं, विटामिन ए, विटामिन बी, विटामिन सी, विटामिन ई, ओमेगा 3 फैटी एसिड्स और इस तरह से बहुत सारे अच्छे बायोकेमिकल्स इनमें हैं जो कि हमारी हेल्थ के लिए एसेंशियल हैं। तो जैसे कि मैंने बताया कि बहुत सारे मरीन एल्गी पे तो काम चल ही रहा है, लेकिन हमें फ्रेश वाटर एल्गी पे भी इस बारे में सोचना है। तो मरीन एल्गीज में है ये अल्वा, पोरफायरा, लैमिनेरिया, क्लोरेला छोड़ दें, क्लोरेला फ्रेश वाटर है, ग्रेसिलेरिया, गैलिडियम, ये सभी मरीन एल्गी हैं और मरीन एल्गी एज ए फूड बहुत अच्छे सब्स्टिट्यूएंट के रूप में सामने आ रही हैं। मैनी एल्गल प्रोडक्ट्स आर यूज्ड इन फूड इंडस्ट्री, जैसे अगर जो हमारा कल्चर मीडिया है उसको फॉर्मेशन के लिए हमें अगर की ज़रूरत पड़ती है, कैराजिनिन की ज़रूरत पड़ती है, तो ये सभी का इनका जो प्रोडक्शन है वो पॉसिबल है। अब आज के समय में जो एल्गी हैं उसको फूड डाई या फूड कलरेंट्स के रूप में भी बहुत अच्छे रूप में ये सामने आ रही हैं, क्योंकि इनकी न्यूट्रिशनल वैल्यू बहुत है। जैसे हमारे यहाँ आइसक्रीम्स हैं या बहुत सारे हमारे फूड प्रोडक्ट ऐसे होते हैं जो कि कलरफुल होते हैं, तो अट्रैक्टिव होते हैं और बच्चे उन्हें खाना पसंद करते हैं। लेकिन हमारे जो पेरेंट्स हैं वो बहुत अलर्ट होते हैं, तो वो कहते हैं नहीं नहीं इसमें कलर पड़ा है और ये हेल्थ के लिए ठीक नहीं होगा और इसे आपको नहीं खाना है, वो बच्चों को नहीं देते हैं। लेकिन आज एल्गी हमारे लिए बहुत सबसे बेस्ट सोर्स है बहुत अच्छे कलर्स के लिए, ब्राउन कलर के लिए, ब्लू कलर के लिए और रेड कलर के लिए। और ये इससे जो कलर्स या डाई ऑब्टेन हो रहे हैं हमें, वो नॉन टॉक्सिक है, उनकी न्यूट्रिशनल वैल्यू भी बहुत ज़्यादा है और वो बहुत ही अच्छे हमारे लिए यूज़ के पदार्थ हो रहे हैं और ये नॉन टॉक्सिक हैं, तो हम इन्हें बिना झिझक के अपने किसी भी फूड मटेरियल को कलर करने के काम में ला सकते हैं। खास करके आइसक्रीम्स वगैरह में हम इन्हें यूज़ कर सकते हैं। तो जैसे रेड कलर है, तो यहाँ पे मैंने शो किया है पोरफायरिडियम प्रुएंटम जो है वो रेड कलर उससे ऑब्टेन हो रहा है। इसी तरह से स्पाइरुलिना प्लेटेंसिस से ब्लू कलर ऑब्टेन हो रहा है। तो इस तरह ये जो स्पाइरुलिना प्लेटेंसिस से ब्लू कलर मिल रहा है, ये फाइकोसाइनिन की प्रेजेंस के कारण। ये तो खैर एक आप चीज़ जानती हैं। तो जितने भी मिल्क प्रोडक्ट्स हैं, मिल्क शेक हैं, आइसक्रीम है, च्युइंगम है, सॉफ्ट ड्रिंक्स हैं, अल्कोहल ड्रिंक्स हैं, इन सभी में ये कलर देने का काम करते हैं और ये नॉन टॉक्सिक हैं, तो हम इनको बिना डरे हम इन्हें यूज़ कर सकते हैं। एल्गी एज ऑर्गेनिक फर्टिलाइजर और बायो फर्टिलाइजर्स के रूप में तो साइनोबैक्टीरिया के बारे में अब हम लोग सभी जानते हैं कि जो साइनोबैक्टीरिया उनको हम एज ए बायो फर्टिलाइजर्स यूज़ करते हैं। तो खास करके राइस फील्ड में इनका बहुत अच्छा उपयोग हो रहा है और उससे प्रोडक्टिविटी काफ़ी इंक्रीज होती है। करीब 30 to 40% इंक्रीज साइनोबैक्टीरिया से प्रोडक्टिविटी में मिलता है। लेकिन ऑर्गेनिक फर्टिलाइजर भी हम एल्गी से बना सकते हैं, ये पहली बार सामने आया है और इसमें ज़्यादातर जो मरीन एल्गी हैं उनसे ब्राउन और रेड एल्गी से उनसे ऑर्गेनिक फर्टिलाइजर्स का भी प्रोडक्शन हो रहा है। तो एल्गी अपने आप में बहुत 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 यूजफुल है और बहुत ही ज़्यादा इको फ्रेंडली है। जैसे कि अब मेरी नेक्स्ट स्लाइड में आपको पता चल रहा है कि ये अल्गल बायोप्लास्टिक्स के रूप में भी इनको यूज़ किया जा सकता है। अल्गल बायोप्लास्टिक्स क्या हैं? अल्गल बायोप्लास्टिक्स जो हैं वो एल्गी से ऑब्टेन पॉलीमर्स हैं जो कि प्रोटीन और कार्बोहाइड्रेट बेस्ड होते हैं और इनका जो डिग्रेडेशन है वो बहुत ही आसान है। तो ये जो पॉलीमर्स हैं इनसे बनने वाला जो प्लास्टिक है वो बहुत ही इको फ्रेंडली है, बायोडिग्रेडेबल है और सस्टेनेबल ग्रीन इनिशिएटिव टू कॉम्बैट प्लास्टिक पोल्यूशन। जो वाइट पोल्यूशन अब आजकल इसे एक हमने नाम दे रखा है कि ये वाइट पोल्यूशन में बहुत ही ज़्यादा हेल्पफुल हैं। तो वाइट पोल्यूशन में ये किस तरह से हेल्पफुल हैं कि बहुत सी एल्गी को ये पाया गया है कि जो हम प्लास्टिक्स जो हैं हम एनवायरनमेंट में फेंक देते हैं, उन पर बहुत सारी एल्गी जो हैं वो अटैच हो रही हैं, उन पर अटैच होकर और उन्हें धीरे धीरे उन्हें बायोडिग्रेड कर रही हैं। तो नॉर्मली जो केमिकल प्लास्टिक हमारे पास है उसको भी डिग्रेडेशन में एल्गी सामने आ रही है, और उसके अलावा एल्गल बायोप्लास्टिक का भी प्रोडक्शन अब रिसेंटली होने लगा है। और आगे आने वाले कुछ सालों में हो सकता है कि हम जो केमिकल प्लास्टिक है उसको हम हटा दें और हम अलगल बायोप्लास्टिक को अपने यूज़ में लें, क्योंकि प्लास्टिक आफ्टर ऑल हमारे लिए बहुत कन्वीनियंट है, बहुत यूज़फुल है, और हमें कहीं पर भी उसको यूज़ करना बहुत ही आसान है। तो हम चाहेंगे कि इस तरह का प्लास्टिक हमारे सामने आए जो कि बायोडिग्रेडेबल हो, और उसमें अलगल बायोप्लास्टिक जो है वो काफ़ी यूज़फुल है। एल्गी एज ए सोर्स ऑफ फार्मास्यूटिकल्स, मैंने आपको अभी बताया कि एल्गी में बहुत सारे विटामिन हैं, बहुत ही अच्छे प्रोटीन्स हैं, कार्बोहाइड्रेट्स हैं, यहाँ तक कि ओमेगा 3 भी है जो कि आपको फिश कॉड लिवर ऑयल में मिलता है, वो ओमेगा 3 भी बहुत सी एल्गी में है। तो एल्गी रिसेंट या बहुत अच्छे फार्मास्यूटिकल के रूप में भी आ रही, बहुत सारी कैल्शियम प्रोडक्ट्स में, मैग्नीशियम प्रोडक्ट्स में, ओमेगा 3 प्रोड्यूस में ये एल्गल सोर्स को यूज किया जा रहा है। एल्गी को इतना डिफाइन करने का मेरा ये मकसद है कि आप लोग एल्गी की तरफ ट्रैक हो। एल्गी बहुत ही यूजफुल है, बहुत जगह पर यूजफुल है। जैसे आजकल हम एल्गल फ्यूल पे भी जा रहे हैं। एल्गल फ्यूल आते हैं बायोफ्यूल। बायोफ्यूल्स मतलब कि हम लिविंग बींग से, लिविंग प्लांट से का यूज़ करें बायोफ्यूल्स के लिए, और उनसे हम क्योंकि ये जो बायोफ्यूल्स हैं ये बहुत से ऐसे फ्यूल्स हैं जो कि इको फ्रेंडली हैं, बहुत ज़्यादा स्क्रीन शेयर हो रही है। एक्चुअली बीच में कुछ प्रॉब्लम आ गया था। ओके, तो एल्गल फ्यूल्स पे अपन बात कर रहे थे कि एल्गल फ्यूल्स क्या हैं। तो एल्गल फ्यूल्स में बायोडीजल, बायोब्यूटेनॉल, बायोगैसोलिन, मीथेन, ये सभी का प्रोडक्शन अब आज के समय में संभव हो रहा है, और इसमें ड्यूनेलिया, क्लोरेला एल्गी का ज़्यादातर यूज़ कर रहे हैं। तो बहुत सारे एल्गल फ्यूल्स हैं कि खास करके बायोब्यूटेनॉल जो है 85% जो है वो इको फ्रेंडली है। बहुत मतलब अब हमें मैक्सिमम इस कुछ आने वाले वर्षों में हमें ये देखना पड़ेगा कि अलगल फ्यूल पर हमें आना पड़ेगा, और अलगल फ्यूल जो है वो ज़्यादा इको फ्रेंडली हैं, ज़्यादा इंटरेस्टिंग है, तो हम इन्हें एज ए अलगल फ्यूल भी यूज़ कर सकते हैं। फिर एल्गी एज बायो रेमेडिएटर्स, ये बहुत सारी एल्गी जो है वो पोल्यूशन कंट्रोल में काम करती हैं, और पोल्यूशन को कम करने का अभी ताजुद्दीन सर ने भी जैसे कि बताया, और इससे पहले के जो स्पीकर हैं उन्होंने भी ये कहा कि हम अपनी जो नदियां हैं उनको रिस्टोर कर सकते हैं। कैसे? क्योंकि वहाँ पर एल्गी जो है बायो रेमेडिएटर का काम करेगी, वहाँ पर जो भी पोल्यूशन है उस पोल्यूशन को रिमूव करने का काम करती है, खास करके हैवी मेटल्स को रिमूव करने का काम करती है एल्गी, और उसके कारण जो है ये बायो रेमेडिएटर्स का भी काम करती है। देन अलगल कल्चर, इस पर अपन थोड़ा डिटेल में बात करेंगे। अलगल कल्चर क्यों करना है? क्योंकि अलगी का जैसे अभी ताजुद्दीन सर ने भी बहुत सारा हमें बताया कि साइटोलॉजिकल या जेनेटिकल प्रोस्पेक्ट्स में, या फिजियोलॉजिकल, मॉर्फोलॉजिकल प्रोस्पेक्ट्स में, या इकोलॉजिकल प्रोस्पेक्ट्स की स्टडी करने के लिए हमें अलगी को कल्चर करना पड़ता है। जैसे उन्होंने आरएनए और डीएनए के लिए बताया, तो उसके लिए पहले हम अलगल कल्चर करते हैं, और उसके बाद ये सारी स्टडीज़ में हमको अलगल कल्चर हेल्पफुल होता है। तो ये तो मैंने एक थोड़ा सा जनरल नॉलेज आपको दिया कि व्हाई एल्गी, why we take algae for a research topic. Then now what is the process of research? Research की क्या process है? Faculty members बहुत से जानते भी हैं, और बहुत जिन्होंने अभी PhD नहीं किया तो हो सकता है कि वो इस process को नहीं जानते हैं, इसलिए थोड़ा सा मैंने एक शॉर्ट में रिसर्च प्रोसेस यहाँ पर लिया है। रिसर्च प्रोसेस क्या है जैसे इसमें मैंने कहा है कि पहले डिफाइन रिसर्च प्रॉब्लम। सबसे पहले तो आपको क्या करना है ये आपके मन में कंसेप्ट क्लियर होना चाहिए, कौन सी रिसर्च प्रॉब्लम लेना है। आपके एरिया को आपको स्टडी करना चाहिए और वहाँ पर पोल्यूशन से रिलेटेड प्रॉब्लम है या उसके अलावा भी कोई भी प्रॉब्लम हो सकती है, उसका व्हाई वाला क्वेश्चन का जवाब आपको देते हुए आना चाहिए। तो व्हाई का क्वेश्चन आप जवाब दे रहे हैं, तब फिर आप उसको डिफाइन कर लेंगे आपकी रिसर्च प्रॉब्लम को। देन जहाँ आपने एक प्रॉब्लम आपने सॉर्ट आउट कर लिया कि हाँ मुझे इस पर वर्क करना है, देन अब आपको क्या करना है, उससे रिलेटेड रिव्यू लिटरेचर, आपको बहुत सारा उस पर रिव्यू लिटरेचर स्टडी करना होगा। रिव्यू कंसेप्ट्स एंड थ्योरीज, मतलब आपको जो भी कंसेप्ट्स और थ्योरीज उस प्रॉब्लम से रिलेटेड है उस पर रिव्यू पढ़ना है, और उसके अलावा रिव्यू प्रीवियस रिसर्च फाइंडिंग, उससे पुराने पहले जो भी फाइंडिंग्स हुई हैं उसके बारे में आपको जानना होगा। अब ये सब जानने के बाद आपको ये कंसेप्ट क्लियर हो जाएगा कि आपने जो टॉपिक लिया उस पर इतना काम हो चुका है और इससे आगे मुझे क्या करना है। तो उसके आगे आपको जो भी करना है उसमें आपके थर्ड स्टेप है, आप फॉर्मुलेट करेंगे हाइपोथेसिस को। और जब आपने एक हाइपोथेसिस फॉर्मुलेट कर लिया कि आपको अभी ये वर्क करना है और इतने पोर्शन पे करना है, और उसमें आप खास करके साइट भी डिसाइड कर लेंगे, और डिसाइड करने के बाद अब आप रिसर्च डिज़ाइन करेंगे। तो रिसर्च डिज़ाइनिंग में आपको ये करना है, प्रोसेस करना है कि आपको करना क्या क्या है, शुरुआत कहाँ से करना है और ख़त्म कहाँ करना है, आपका एक ये कंसेप्ट माइंड में क्लियर होना चाहिए। देन उसके बाद अब आप कलेक्शन ऑफ डाटा करेंगे। कलेक्शन डाटा जैसे ही कलेक्ट हो गया उन पॉइंट से रिलेटेड जो आपने रिसर्च डिज़ाइन किया है उससे रिलेटेड जब डाटा कलेक्ट हो जाएगा तो उस डाटा को आप एनालाइज करेंगे, और उस एनालिसिस के आधार पे आप अपनी उस रिजल्ट को इंटरप्रेट करेंगे, उस एनालिसिस को इंटरप्रेट करेंगे और रिपोर्ट करेंगे कि आपको क्या आपने एनालाइज किया है उस प्रोसेस में। तो एल्गी के या हम साइंस के केस में देख लें कि किसी भी केस में बेसिक रिसर्च भी होती हैं, अप्लाइड रिसर्च भी होती हैं। आजकल ज़्यादातर जो वर्क हो रहा है अप्लाइड रिसर्च पे हो रहा है, आपको भी अप्लाइड रिसर्च पे लेना चाहिए, लेकिन ये मेरा कहना बिल्कुल भी नहीं कि बेसिक रिसर्च गलत है। मैंने आपसे कहा भी पहले भी कि बेसिक रिसर्च ही हमको एक बेस देती है, हमको एक स्टेप देती हैं कि हमको इस तरह से इसके ऊपर वर्क करना है। हमारे लिए हमारा बहुत सी जो फ्लोराज हैं, अलगल फ्लोराज हैं या अलगल वेजिटेशन है जिनके बारे में हम नहीं जानते, तो हम उनके बारे में भी सोच सकते हैं। तो एक ये फंडामेंटल या प्योर रिसर्च है, और प्योर रिसर्च भी अपने आप में एक बहुत ही मोटिवेशनल रिसर्च होती है, इसमें कोई शक नहीं। आजकल जो मैंने कहा कि अप्लाइड रिसर्च पे काम हो रहा है, अप्लाइड रिसर्च पे कि जो भी बेसिक डेटाज हमारे पास पहले से हैं उनके आगे अब हमको उनकी साइंटिफिक जो हमारे एरिया की जो भी प्रॉब्लम है तो उसको हम अप्लाइड रिसर्च करके हम उसे क्योर कर सकते हैं। जैसे बहुत सारी प्रॉब्लम्स हमारी होती हैं, जैसे दो स्टेम्स मिलकर वो भी उस रिसर्च को कर सकते हैं। जैसे बॉटनी और जूलॉजी मिलकर अभी एक थीसिस आई थी, तो उसमें फिश पर उन्होंने हल्दी का काम किया था कि हल्दी के थ्रू उसमें बहुत सारी उन्होंने डिसीजेस को दूर करने में हेल्प मिली थी। तो इस तरह से हम अप्लाइड रिसर्च में बहुत सा वर्क कर सकते हैं जो कि हमारी जो डेली की प्रॉब्लम्स हैं, हमारी जो आसपास की प्रॉब्लम्स हैं, उन प्रॉब्लम्स को सॉल्व कर सकते हैं और उन प्रॉब्लम को स्टेप वाइज हम अलग कर सकते हैं। फिर कोरिलेशनल रिसर्च, इसके बारे में भी आप जानते हैं। कोरिलेशनल रिसर्च क्या है? कोरिलेशनल रिसर्च है कि हम मान लीजिए हम दो पॉन्ड ले लें और दोनों पॉन्ड की हम एल्गल फ्लोरा को स्टडी करें और उनका कोरिलेशन बताएं, उनमें से कौन एल्गा जो है वो ज़्यादा बेटर है, वहाँ का पानी ज़्यादा सेफ है, या वहाँ पर वो पॉल्यूशन को रिमूव करने का काम कर रही है, और दूसरे पॉन्ड में जो एल्गा है वो पॉल्यूशन कंट्रोल नहीं कर पा रही। तो हम कोरिलेशनल रिसर्च कर सकते हैं। डिस्क्रिप्टिव रिसर्च ये ज़्यादातर आर्ट्स में होती है, साइंस में भी होती है, लेकिन अभी इसकी मात्रा हमारे यहाँ कम है। और एक्सपेरिमेंटल रिसर्च हम लोग ज़्यादा करते हैं। जो भी हमने अपना रिसर्च टॉपिक लिया है उस पर एक्सपेरिमेंट्स के द्वारा उसको हम रिजल्ट्स उसके ड्रॉ करते हैं। क्वांटिटेटिव रिसर्च और क्वालिटेटिव रिसर्च ये दो टाइप्स ऑफ रिसर्च हैं जो कि दोनों का कॉम्बिनेशन भी हो सकता है और दोनों को हम अलग भी कर सकते हैं। ज़्यादातर हम लोग कॉम्बिनेशन ही करके चलते हैं कि क्वालिटेटिव रिसर्च में हम नॉर्मली मैथमेटिक नहीं होता, पर हम उसमें ज़्यादातर जो है वो अपने बिलीव्स और मीनिंग्स का इस्तेमाल करते हैं कि खास करके किसी एरिया में जो है वो कौन सी एल्गा बहुत ज़्यादा मात्रा में है और वहाँ पर वो ज़्यादा मात्रा में होकर और वहाँ पर वो क्या कर रही है, पॉल्यूशन कर रही है या पॉल्यूशन को दूर करने का काम कर रही है, तो ये क्वालिटेटिव रिसर्च में आएगा। लेकिन क्वांटिटेटिव रिसर्च में जब हम वहाँ पर मैथमेटिकल डाटाज लगा दें, हम स्टैटिस्टिकल डाटा या मैथमेटिकल डाटा या कंप्यूटेशनल टेक्निक्स यूज़ कर लें, तो दैट इज़ क्वांटिटेटिव रिसर्च। अब ये सब आपने एक हाइपोथेसिस कर लिया। अब आपके माइंड में जो एक रिसर्च क्वेश्चन पैदा हुआ था वो एकदम से क्लियर होना चाहिए। आपको माइंड में आपका कंसेप्ट क्लियर होना चाहिए कि आपको काम क्या करना है। आपका फोकस होना चाहिए, फोकस्ड होना चाहिए वो क्वेश्चन, कॉन्साइज होना चाहिए। मतलब आपका ऐसा नहीं कि आप बहुत बड़े एरिया में ले लें और इतना कि काम बढ़ा लें कि आप उस काम को न कर पाएँ। तो कॉन्साइज भी होना चाहिए आपका एरिया, आपका काम, कि आप मतलब 3 साल में या 4 साल में या 5 साल में उस काम को कंप्लीट कर लें। और कॉम्प्लेक्स होना चाहिए। कॉम्प्लेक्स मतलब कि इतना भी इजी नहीं होना चाहिए कि वो ऐसा ना लगे कि ये पीएचडी थीसिस के लायक टॉपिक ही नहीं था, ये तो एमएससी के लायक टॉपिक था और आपने ले लिया है। तो आपका जो टॉपिक है वो थोड़ा कॉम्प्लेक्स भी होना चाहिए और ऑर्ग्यूएबल मतलब कि आप उस टॉपिक पर से रिलेटेड जितने भी क्वेश्चंस हैं उनके आप आंसर्स कर सकें, आर्ग्यू कर सकें, आप उसकी जवाब दे सकें। तो ये आपकी स्टेप्स में आना चाहिए कि आपकी जो थीसिस है वो कैसे आप उसको बेटर बनाएंगे। अब आपने एक कंसेप्ट क्लियर कर लिया आपको क्या काम करना है और कैसे करना है। अब आप क्या करेंगे, उससे पहले उससे रिलेटेड जो रिव्यूज़ हैं उनको, लिटरेचर रिव्यू हैं उनको आपको स्टडी करना होगा। उससे क्या होगा कि आपके दिमाग में एक कंसेप्ट क्लियर हो जाएगा कि कितना काम हुआ है और किस तरह का काम हुआ है। तो आप अपनी जो प्रॉब्लम आपने डिटेक्ट की है उस प्रॉब्लम से रिलेटेड जितने भी लिटरेचर्स हैं उनको आपको स्टडी करना चाहिए। अब जो ये लिटरेचर रिव्यू है आपके इन सभी क्वेश्चंस का जवाब देना चाहिए। इस लिटरेचर रिव्यू का पहला जैसे व्हाट आर द मेजर इश्यूज एंड डिबेट अबाउट द रिसर्च प्रॉब्लम। जो आपने रिसर्च प्रॉब्लम ली है उससे में मेजर जो इश्यूज और डिबेट किस बारे में है वो आपको पता होना चाहिए। आर देयर एनी गैप्स इन नॉलेज ऑफ द सब्जेक्ट, यदि जो सब्जेक्ट है उसके नॉलेज में कुछ गैप आ गया है तो वो गैप क्या है और हाउ कैन आई ब्रिज द गैप, हम उस गैप को कैसे ख़त्म करेंगे। व्हाट डायरेक्शंस मेथोडोलॉजी आर इंडिकेटेड बाय द वर्क ऑफ अदर रिसर्चर, जो दूसरे रिसर्चर्स हैं वो एक आपको डायरेक्शन बता रहे हैं कि हमने किस डायरेक्शन में काम किया है, तो उस डायरेक्शन में ही आप भी जा सकते हैं या उसके अपोजिट भी अगर आपको सही लग रहा है तो आप उसके अपोजिट डायरेक्शन में भी काम कर सकते हैं। तो लिटरेचर रिव्यू से आपके दिमाग में ये कंसेप्ट क्लियर होंगे। What are the key theories, concept and ideas known about the subject? सब्जेक्ट के बारे में क्या की आइडियाज़ लिए हैं या की थ्योरीज लिए हैं, क्या कंसेप्ट लिए हैं, क्या आइडियाज़ लिए हैं, ये आपको कंसेप्ट क्लियर होगा। व्हाट इज़ द क्रोनोलॉजी ऑफ द डेवलपमेंट ऑफ नॉलेज, मतलब फर्स्ट ऑर्डर में क्या रिसर्च हुआ, फिर उसके बाद आज के समय में मतलब पहले से लेकर आखिरी तक में अभी तक आपके टॉपिक पर क्या डेवलपमेंट हुई है, ये आपके सामने आ जाना चाहिए। वेदर द रिसर्च क्वेश्चन ऑलरेडी हैज़ बीन आंसर्ड बाय समवन एल्स, आपको ये भी देखना है कि आपका जो वेदर रिसर्च क्वेश्चन है वो किसी दूसरे ने सॉल्व किया हुआ ना हो। आपका क्वेश्चन ऐसा होना चाहिए कि जो किसी ने भी सॉल्व नहीं किया है और अब आप उसको सॉल्व करने जा रहे हैं। तो यूनिकनेस बहुत ज़रूरी है आपके वर्क की। अगर आप यूनिक वर्क कर रहे हैं तो वो आपके लिए भी इंटरेस्टिंग होगा और आपके जब पीएचडी वाइवा होगा तो सामने जो एक्सटर्नल होंगे उनके लिए भी वो बहुत ही अच्छा साबित होगा। डेवलपमेंट ऑफ वर्किंग हाइपोथेसिस, अब आपको आपने लिटरेचर भी पढ़ लिया, आपने सब कर लिया, अब आपको एक वर्किंग हाइपोथेसिस बनाना है कि आपको काम कैसे शुरू करना है, अब कहाँ से शुरू करना है और कहाँ ख़त्म करना है। दैट इज डेवलपमेंट ऑफ वर्किंग हाइपोथेसिस। After extensive literature survey, researchers should state in clear terms the working hypothesis. For a researcher's hypothesis is a formal question that he intends to resolve a hypothesis is a proposed explanation for an observable phenomenon which is capable of being tested by scientific method. तो ये एक कंसेप्ट हमारे दिमाग में क्लियर होना चाहिए कि जो हाइपोथेसिस अब आप वर्किंग हाइपोथेसिस बना रहे हैं वो आपके क्वेश्चन को कैसे सॉल्व करेगा, किन मेथड से सॉल्व करेगा। तो अब आता है मेथोडोलॉजी। मेथोडोलॉजी इन केस ऑफ एल्गी के लिए मेथोडोलॉजी बहुत इजी है कि आप उसका कलेक्शन करें, आप उनकी स्लाइड्स बनाएं। टैक्सोनॉमी अगर आप कर रहे हैं तो टैक्सोनॉमिकल में आप उसकी स्लाइड बनाएंगे। तो मेथोडोलॉजी भी अब आपका, क्योंकि आपने लिटरेचर स्टडी किया है तो बहुत सारी चीज़ें अब आपके दिमाग में क्लियर हैं। तो आप एक रिसर्च डिज़ाइन बनाएंगे कि हमको स्टेप वाइज क्या करना है, क्या हमें कलेक्शन कब करना है, आइसोलेशन कब करना है, फिर हमें उनकी स्लाइड्स बनाना है या हमें उसका कल्चर करना है, वो किस तरह से स्टेप वाइज करना है, ये हमें मेथोडोलॉजी के अंतर्गत पढ़ना है। डाटा कलेक्शन मेथोडोलॉजी आपने किया, मेथोडोलॉजी का ही ये एक स्टेप है कि अब आपको डाटा कलेक्शन करना है कि डाटा किस तरह से एल्गी से रिलेटेड आपने जो भी टॉपिक लिया है, यदि वो क्वालिटेटिव है या क्वांटिटेटिव है, तो उस हिसाब से आपको डाटा कलेक्शन करना है। और डाटा किस तरह का चाहिए ये भी आपको देखना है। अगर आपको क्वांटिटी में डाटा चाहिए तो उसकी भी स्टडी करना होगी, और यदि आपको क्वालिटी में डाटा चाहिए तो उसकी भी स्टडी आपको करना होगी। तो एल्गी में जैसे कि मैंने आपको कहा कि आपने पहले एल्गी का कलेक्शन किया, आइसोलेशन किया, फिर आप उसका स्लाइड फॉर्मेशन करते हैं। नॉर्मल एल्गी जो है, खास करके फ्रेश वाटर जो एल्गी हैं या मरीन एल्गी हैं, उनका भी जो स्लाइड फॉर्मेशन है वो बहुत ईजी है। लेकिन जो बैसिलरोफाइट्स हैं, जो डायटम्स हैं, इनमें स्पेशल मेथड हमको अप्लाई करना पड़ती है। क्यों? क्योंकि इनके ज़्यादातर जो डायटम्स हैं वो पॉल्यूटेड वाटर में होते हैं, और पॉल्यूटेड वाटर में होने के कारण इनके ऊपर इम्प्योरिटीज़ बहुत ज़्यादा होती हैं, और वो इम्प्योरिटीज़ जब तक अगर हम रिमूव नहीं करेंगे तब तक हमको इसका सही डायमेंशंस नहीं मिलते, सही हमको इनका डिज़ाइन नहीं मिलता, या हमें इनके बारे में सही जानकारी नहीं होती। तो हमको माउंटिंग ऑफ डायटम्स कैसे करना है उसकी प्रोसीजर मैंने यहाँ दी है। कि डायटम्स क्या हैं? डायटम्स यूबिक्विटस है, मतलब सर्वव्यापी, हर जगह पर मिलते हैं। कलेक्टेड बाय प्लैंकटन नेट, हमेशा इनको हम प्लैंकटन नेट से ही कलेक्ट कर सकते हैं। To cleaning and mounting of diatoms, following method is applied so that contents on cell or deposits on silicified cell wall may destroy. जो डायटम्स की सेल वॉल है, जैसे कि ये डायग्राम में आप देखिए, इनमें बड़े सुंदर इनके डायग्राम्स होते हैं, बहुत सुंदर इनके शेप्स होते हैं। वो किस कारण से होते हैं? क्योंकि इनकी वॉल जो है सिलिसिफाइड होती है, सिलिका की बनी होती है, और सिलिका के बने होने के कारण इन पर ये अलग अलग डिजाइन आ जाते हैं। इनमें स्पाइंस टाइप कांटीनुमा रचनाएं भी होती हैं। तो ये जो डिजाइन्स हैं, जब तक हम इनके बारे में इनका डिटेल हम नॉलेज नहीं कर पाएंगे, जब तक कि ये इनके ऊपर से इम्प्योरिटीज हम ना हटा लें। तो इसके लिए हमें क्या करना है वो मेथड मैं यहाँ पर आपको बता रही हूँ, क्योंकि थोड़ा सा एक ये रेयर मेथड है, हो सकता है आपको नॉलेज नहीं होगा, इसलिए मैंने यहाँ पर ये मेथड डायटम्स की रखी है। टू अ पोर्शन ऑफ मटेरियल एड 20 ml कॉन्सेंट्रेटेड HCl इन अ बीकर। उसके बाद आपको बीकर को शेक करना है, और शेक करना है किसमें? सेंट्रीफ्यूज मशीन में शेक करना है, और करीब 15 से 20 बार उसको डिस्टिल वाटर से वॉश करना है, वॉश करके फिर शेक करना है। इस तरह से बहुत बार आप उसको साफ करेंगे ना। अब उससे क्या होगा कि जब सेंट्रीफ्यूज मशीन आप चलाएंगे तो उसमें जो डायटम्स हैं वो बीकर में नीचे सेटल डाउन हो जाएंगे। उनको आपको फाइन क्लॉथ से सैंड पार्टिकल्स हटाते हुए उनको आपको स्टीम कर लेना है। पोर ऑफ एक्सेस वाटर हटा के अब उसमें बहुत कम मात्रा में H2SO4 डालना है। H2SO4 सिलिका को डिजोल्व करने का काम करता है, इसलिए उसमें H2SO4 डालना होता है। जो कवर स्लिप पे ही ये पूरी प्रोसेस करते हैं अपन, और फिर उस कवर स्लिप को आपको फ्लेम पर गर्म करना है। स्लाइडली फ्लेम पर गर्म करने पे क्या होगा? सो दैट ऑर्गेनिक मैटर डिजोल्व, H2SO4 जो है ऑर्गेनिक मैटर को डिजोल्व कर देगा। फिर अब क्या करना है, पोटेशियम डाइक्रोमेट क्रिस्टल्स ऐड करना है, जिसके कारण उनका कलर ग्रीन हो जाएगा। और ग्रीन कलर, क्योंकि उनका नॉर्मली जो आप HCl वगैरह यूज करते हैं तो उनका जो ओरिजिनल कलर है वो चला जाता है, इसलिए हमको पोटेशियम डाइक्रोमेट डालना होता है ताकि वो वापस से अपने ओरिजिनल कलर पे आ जाए। एग्जामिन इट अंडर माइक्रोस्कोप, इफ एनी कंटेमिनेशन देयर, देन रिपीट अबाउट प्रोसीजर अगेन। क्योंकि ये जो प्रोसीजर है HCl और H2SO4 दोनों ही एसिडिक हैं, और इनमें क्या होता है कि कई बार ये बहुत अच्छे से वर्क करते हैं, कई बार हो सकता है कि आपका जो एसिड है वो अगर डाइल्यूटेड है तो वो वर्क नहीं कर पाएगा। तो ये हमें माइक्रोस्कोप में बार बार इन्हें देखना पड़ता है इस प्रोसीजर के बाद, और जब अगर आपको कंटेमिनेशन मिल रहा है तो इस प्रोसीजर को फिर आपको अगेन करना है, बार बार करना है। 1 या 2 बार में हो जाता है, वैसे ज़्यादा प्रॉब्लम नहीं आती। अब उसके बाद जैसे आपने ये पूरी प्रोसीजर कर लिया, आपने देख लिया कंटेमिनेशन नहीं है, तब आप इसको कई बार डिस्टिल वाटर से धोते हैं। आफ्टर क्लीनिंग, thrown away supernatant water, now place a small amount of cleaned item on cover slip and add a drop of alcohol. कवर स्लिप पे बहुत ही थोड़ा मात्रा में आपको डाइटम लेना है और उसमें अल्कोहल लेना है, उससे अल्कोहल को फिर गर्म करना है कवर स्लिप पर, so that the item is spread evenly on cover slip. Change the material of the item 2 times in Zalol and mount in Canada box. बहुत इजी मेथड है, बहुत अच्छी मेथड है, लेकिन अगर प्रॉपर मेथड होती है तो डाइटम बहुत ही सुंदर रूप में हमारे सामने आता है। यदि हम प्रॉपरली नहीं कर पाते हैं तो थोड़ा सा उसकी डायग्राम्स, जैसे यहाँ पे आप देख रहे हैं ये डाइटम्स की डायग्राम है, यहाँ पर आपको दिख रहे हैं, तो वो जो ये डाइटम्स हैं ये आपको क्लियरली तभी दिखेंगे जब हम इस प्रोसीजर को अप्लाई करेंगे। अब इसके बाद ये कल्चर मेथड के बारे में अब मैंने आपको बताना चाहे, वैसे ताजुद्दीन सर ने बहुत डिटेल में आपको बताया, उसको मैं थोड़ा सा शॉर्ट में भी आपको बताती चलती हूँ कि कल्चर कंडीशंस एल्गा के लिए हमको ये रिक्वायरमेंट्स हैं, वाटर, कार्बन डाइऑक्साइड, मिनरल्स, लाइट, टेम्परेचर। देन कल्चर माइक्रो एल्गी का, माइक्रो एल्गी में ये जैसे ब्लू ग्रीन एल्गी आती हैं, ग्रीन एल्गी आती हैं, इसके अलावा बैसिलोफाइटा, ज़ैंथोफाइटा के मेंबर्स जो है माइक्रो एल्गी में आते हैं। तो माइक्रो एल्गी में यूनिसेलुलर एल्गी भी हैं और कुछ फिलामेंटस एल्गी भी हैं जो जिनको हम माइक्रो एल्गी में रखते हैं। The size ranges from a few micrometer to a few hundred micrometers, include freshwater and marine forms. माइक्रो एल्गी में दोनों फॉर्म्स आ सकते हैं, फ्रेश वाटर भी और मरीन भी, क्योंकि जो समुद्री हैं लेकिन यूनिसेलुलर हैं तो वो भी माइक्रो एल्गी के अंतर्गत ही हम स्टडी करते हैं। कलेक्शन ऑफ एल्गी, एल्गी का कलेक्शन जो है, क्योंकि एल्गी जो है वो ज़्यादातर हमको ब्लूम फॉर्मिंग होती है या स्कम फॉर्मिंग होती है, तो स्कम या ब्लूम के रूप में है, तो हम उसको कैसे कलेक्ट करेंगे, तो इसके लिए हमको वाटर सैंपल्स कैन बी कलेक्टेड इन स्मॉल कंटेनर्स एंड पुट इन एन आइस फिल्ड कूलर। क्या होता है, क्योंकि बहुत सी एल्गी ऐसी होती है जो कि आपको वो माइक्रोस्कोपिक है, और माइक्रोस्कोपिक होने के कारण आप उसको जो है वो किसी भी तरह से आप अपने लैब तक नहीं ला सकते, तो उसका तरीका यही है कि आप उसको एक स्मॉल कंटेनर में डाल लें पानी सहित, वहाँ पर जो भी पानी है उस पानी के सहित ही आप उसमें डाल लें और फिर वो कंटेनर को एक आइस बॉक्स में रख दें। तो उससे क्या होगा कि उसमें जो डिग्रेडेशन की जो प्रोसेस है वो स्लो हो जाएगी। Some algae form scum on the water surface. In such cases, scoop a small sample from the surface. Scum forming है तो वो तो scoop से ही निकल आएगी और आप अपने कंटेनर में रख लेंगे। Floating microalgae can be collected with mesh net, और इसी तरह से a deep water sampler can be used. Okay. A deep water sampler can be used to collect water several meters below the surface. आइसोलेशन ऑफ माइक्रो एल्गी, ये हम पिपेट की सहायता से कर सकते हैं माइक्रोस्कोप में देखते हुए, और आप इसको आसानी से माइक्रो पिपेट से सेपरेट कर सकते हैं, और सिंगल सेल या यूनी एल्गल कल्चर्स के लिए हम इन्हें निकाल सकते हैं। सेंट्रीफ्यूगेशन मेथड, अभी मैंने आपको बताया कि सेंट्रीफ्यूज मशीन से भी हम और वॉशिंग करके भी इनको हम आइसोलेट कर सकते हैं। बाय एक्सप्लॉइटिंग द फोटोटैक्टिक मूवमेंट, इनका हम लैब में इसको कर सकते हैं कि वहाँ पर हम लाइट जला दें और लाइट की तरफ ये फोटोटैक्टिक मूवमेंट करती हैं, और उससे भी हम इनका आइसोलेशन कर सकते हैं। और इसी तरह से सीरियल डाइल्यूशन अगर प्लेट मेथड पे भी हम इसे सेपरेट कर सकते हैं। अब हमने जैसे इन्हें आइसोलेट कर लिया, आइसोलेट करने पर अब हम इन्हें कल्चर करना है, तो इन्हें हम इनोकुलेट करेंगे नीडल से, और नीडल से इनोकुलेट करके इसको हम इंक्यूबेट करेंगे अंडर सुटेबल कंडीशन ऑफ लाइट एंड टेम्परेचर। कुछ समय के बाद ही हमको इनमें अलगल कॉलोनीज डेवलप होती हुई दिखाई देती हैं। इनको हम कल्चर ट्यूब में भी ट्रांसफर कर सकते हैं फॉर प्योर कल्चर। माइक्रो एल्गी के कल्चर के लिए ये और भी मेथड्स हैं। माइक्रो एल्गी कैन बी कल्चर्ड इन फोटो बायो रिएक्टर्स। ये सभी फोटो बायो रिएक्टर्स हैं, जैसे फाइबर ग्लास ट्यूब्स हैं, प्लास्टिक बैग्स हैं, फ्लैट पैनल्स हैं। इन सभी के द्वारा हम इन्हें कल्चर कर सकते हैं आसानी से। कल्चर मीडिया, हर प्रकार के जितने भी एल्गी हैं उनके लिए कल्चर मीडिया भी सेपरेट हैं। अलग अलग कल्चर मीडिया हैं, जैसे कि ग्रीन एल्गी के लिए बोल्स बेसल मीडियम काम में आता है विद विटामिन्स। ये जो बीबीएम मीडियम है, ये खास करके जितने भी ग्रीन एल्गी हैं उनके लिए बहुत ही यूजफुल है। क्यू नंबर 10 मीडियम नाम का ये जो है, ये ब्लू ग्रीन एल्गी के लिए, डायटम्स के लिए और ग्रीन एल्गी के लिए। नॉर्मली मैक्सिमम एल्गी के लिए जो कल्चर मीडिया यूज़ किया जाता है वो क्यू नंबर 10 मीडियम ही यूज़ किया जाता है। क्यू 13 मीडियम भी, इसका भी इस्तेमाल होता है। इसमें कई इसेंशियल मिनरल्स होते हैं, ट्रेस एलिमेंट्स होते हैं दैट आर रिक्वायर्ड बाय एल्गी फॉर ग्रोथ बट विथ नो कार्बन सोर्स, एंड हैज दिस मीडियम इज सुटेबल फॉर ओनली फॉर द ग्रोथ ऑफ फोटोटॉस। जो सनलाइट में होती हैं एल्गी, सिर्फ उन्हीं के लिए ही यूजफुल है ये। और ख़ास करके जो बॉट्रियोकॉकस ब्राउनी है, इसके लिए ये बहुत ही यूजफुल है। मिकेल सॉल्यूशन, ये भी, या जितने भी हैं ये सभी आइटम्स के लिए बहुत अच्छे हैं, और मरीन एल्गी के लिए भी बहुत अच्छे सॉल्यूशन है। जैसे मिकेल सॉल्यूशन फ्रेश वाटर डाइटम्स के लिए, F2 मीडियम जो है ये भी डाइटम्स के लिए और मरीन एल्गी के लिए भी यूज़ होता है। वॉलनेस मीडियम मरीन माइक्रो एल्गी के लिए यूज़ होता है। स्क्रेबर सॉल्यूशन जो है वो डाइटम्स के लिए यूज़ होता है, और TMRL माइक्रो एल्गी के लिए यूज़ होता है। हार्वेस्टिंग ऑफ माइक्रो एल्गी हमने पूरा तरह से कल्चर कर लिया, अब उसे हार्वेस्ट करना है। तो हार्वेस्ट के लिए अब टाइम नहीं है, वरना मैं ये इसका डिटेल में आपको बताती। बहुत से मेथड्स हैं, जैसे कोएगुलेशन एंड फ्लोकुलेशन मेथड है, फ्लोटेशन है, सेंट्रीफ्यूगेशन है, सेडिमेंटेशन है, फिल्ट्रेशन और कॉम्बिनेशन ऑफ वेरियस टेक्निक्स। इस तरह से हम इनका हार्वेस्टिंग भी कर सकते हैं। तो एल्गी अपने आप में एक बहुत ही अच्छा सब्जेक्ट है, बहुत अच्छा वर्क इस पर हो सकता है। और आप चाहें तो हाँ, आप चाहें तो इस पर बहुत ही अच्छा वर्क आप कर सकते हैं, और उस अच्छे वर्क से आप यहाँ पर एक अच्छा मैसेज अपने उसमें दे सकते हैं कि हम एल्गी के थ्रू किस तरह से बेटर वर्क अपने सोसाइटी के लिए कर सकते हैं। तो इस तरह मैंने अपना टॉपिक यहाँ पर फिनिश किया। थोड़ा सा टाइम कम था, नहीं तो मैं और आपको अच्छा डिटेल में बता सकती थी इस बारे में। सो थैंक यू एवरीवन फॉर लिसनिंग मी।
थैंक यू सो मच मैम। किसी की कोई क्वेरी है तो पूछ सकता है। Okay. Now, I would like to invite co-convener of the program, Dr. Manoj Kumar Rawat sir, to deliver vote of thanks to Rashmi ma'am. Dr. Manoj Kumar Rawat.
थैंक यू नितिका मैडम। आयुक्तालय जयपुर राजकीय महाविद्यालय बूंदी के वनस्पति शास्त्र विभाग के द्वारा आयोजित ज्ञान गंगा कार्यक्रम में उपस्थित सभी प्रतिभागी गण, मैं धन्यवाद ज्ञापित करता हूं डॉक्टर रश्मि सिंह मैम को, प्रोफेसर एंड हेड डिपार्टमेंट ऑफ बॉटनी, गवर्नमेंट कॉलेज सतना, जिन्होंने वनस्पति शास्त्र की महत्वपूर्ण शाखा एल्गी, जिसे फाइकोलॉजी और एल्गोलॉजी कहते हैं, आपने शोध जिसमें कल्चर माउंटिंग व इसकी आवश्यकता, जिसमें कई सारे थ्रस्ट एरिया के बारे में बताया आपने, जैसे ऑर्गेनिक फर्टिलाइजर्स, आज इसकी बहुत ही अधिक आवश्यकता है, जो कई सारे पेस्टिसाइड्स किसान लोग काम में ले रहे हैं, जिससे सॉइल पोल्यूशन, लैंड पोल्यूशन बहुत तेजी से हो रहा है। एल्गल बायोप्लास्टिक, आज इसकी भी बहुत अधिक आवश्यकता है, क्योंकि ये इजिली डिग्रेडेबल है, आपने बताया फार्मास्यूटिकल जो कि ओमेगा फैटी एसिड का स्रोत है, एल्गल बायोफ्यूल, इसके बारे में भी जितना कम कहें हम उतनी कम है, क्योंकि बायोफ्यूल आज के समय जो है पोल्यूशन बहुत तेजी से हो रहा है, एयर पोल्यूशन, तो एल्गिल बायोफ्यूल का भी एक बहुत अच्छा थ्रस्ट एरिया बनता है। बायो रेमेडिएटर्स जो कि पोल्यूशन कंट्रोल करने में काम में आता है, आपने इसकी आवश्यकता के बारे में बताया। बहुत बहुत धन्यवाद मैम, थैंक यू।
थैंक यू सर।
थैंक यू सर। पार्टिसिपेंट्स के लिए फीडबैक फॉर्म दे दिया गया है, उसका लिंक कृपया आप इसे भर दीजिए। थैंक यू सो मच।